मेंशन करना जरूरी नहीं था। या खोई चाहूं तुम्हें क्या कसूर है मेरा। Excuse me. बिहान म सुतिरहेको ए मुसा भन्दै थियो त भर्खर खाएको होइन तर पनि के कम भयो चिनीको चिया पकाइकिन दिनु गाम्बु
म त पकाइदिनु है
किन
म त
Hey, hi Baba. I'm so glad you're back. How many hours do you sleep, Ima? I don't know. Maybe I slept 12 hours today. Maybe more. I sleep a lot. Anyways, let's study, okay? I would like to say, hey, welcome back, Baba. Welcome, bro. Welcome. Now, don't you dare to disappear, okay, Baba? Don't you dare to disappear. You use flat pig. What is flat pig? Anterior pituitary hormone. Okay, now I do. I think I will remember it. What is flag pig? Huh? Is it something like flashcards? Mnemonics. Okay. Excuse me. Happy evening. Same to you, Devashish. Now don't dare to be rude, okay? Devashish कुछ ना कुछ हगेगा. I know. पहले भी ऐसा ही करता था ना. Okay, boys, I'm not gonna disappear. Now let's study, okay? 50 मिनट का स्लॉट है ये स्लॉट 1 में लिख भी देती हूँ. SLOT slot 1. Mosquito. Biotechnology principles and processes. Biotechnology deals with techniques of using live organisms or enzymes to produce products and processes using human cells. Okay. Biotechnology, among many, the two core techniques that enable birth of modern biology, genetic engineering, recombinant DNA technology (RDT), the integration of natural science and the integration of natural science and organism, cells, parts thereof, and molecular analog for products and services. The integration of natural science and organism. Okay, this is European Federation Biotechnology definition by European Federation of Biotechnology. The integration of natural science and organism, parts thereof, and molecular analogs for products and services. Principle of biotechnology, genetic engineering, recombinant DNA technology (RDT) to alter the chemistry of genetic material, and bioprocess engineering, genetic engineering, bioprocess engineering. This into host organism and thus change the phenotype of the host organism. Bioprocess engineering, maintenance of sterile, microbial, contamination-free ambience in chemical engineering process to enable growth of only the desired microbes. Eukaryotic cells. Astaghfirullah. I'll kind of die with embarrassment. Eukaryotic cell in astaghfirullah. Desired only the desired microbes. Eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. by process. This change into host organism and thus change the phenotype of the host organism. Phenotype of the host organism. The techniques of genetic engineering, which include creation of recombinant DNA. The techniques of genetic engineering, which includes creation of recombinant DNA, use of gene cloning, and gene transfer overcome this limitation and allows us to isolate recombinant. Creation of recombinant DNA, use of gene cloning and gene transfer overcome this limitation and allow us to isolate and introduce only one or a set of desirable genes without introducing desirable gene into the target organism. Do you know the likely fate of a piece of DNA? In a chromosome, there is a specific DNA sequence called the origin of replication. A specific DNA sequence called the origin of replication. Multiplication of any piece of DNA in an organism, it needs to be part of a chromosomes, which has a specific sequence known as origin of replication. This can also be called as cloning or making multiple identical copies of template DNA. Cloning, making multiple identical copies of template DNA. Multiple identical copies of template DNA. Cloning, template DNA. Multiple copies of template DNA. Multiple copies of template DNA. Okay, multiple copies of template DNA. Astaghfirullah. Template DNA. This can also be called as cloning or making multiple identical copies of any template DNA. First instance, okay, artificial recombinant DNA, Salmonella typhimurium. Bacteria Salmonella typhimurium, Stanley Cohen and Herbert Boyer, 1972. Linking a gene cloning antibiotic resistance with a native plasmid of Salmonella typhimurium. First recombinant DNA, okay, linking a gene encoding antibiotics. linking a gene encoding antibiotics resistance with a native plasmid, Salmonella typhimurium, Stanley Cohen and Herbert Boyer. Molecular scissor, restriction endonuclease enzyme, restriction enzyme, not endonuclease, restriction enzyme. The cutting of DNA at a specific location became possible with the discovery of so-called molecular scissor restriction enzyme. Restriction enzyme. Plasmid act as a vector to transport the piece of DNA attached to it. Okay. The linking of antibiotic resistant gene with the plasmid vector became possible with the enzyme DNA ligase. Gene with the plasmid vector became possible with the enzyme DNA ligase. This makes a new combination of circular autonomously replicating DNA called in vitro. Okay, in vitro DNA created in vitro and is known as recombinant DNA. Like you say, recombinant DNA. Recombinant DNA. This makes a new. This makes a new. And this recombinant DNA. With this DNA is transferred into E. coli, a bacterium closely related to Salmonella. It could replicate using the new host DNA polymerase enzyme. New host DNA polymerase enzyme. New host DNA polymerase enzyme. Also, the ability to multiply copies of antibiotic resistant gene in E. coli was called cloning. Genetically modifying an organism. 3 steps, basic steps. Genetically modifying an organism. Identification of DNA with a desirable gene. Introduction of the identical DNA into the host. Maintenance. Okay. Introduction of the identical DNA into the host. Maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Who knows? 200 strain, 900 restriction enzyme. Tools of recombinant DNA technology. Restriction enzyme, plasmid, vector, host. Restriction enzyme, polymerase enzyme for cloning. Polymerase enzyme, ligase, vector and host organism. Vector, host. Ligase, polymerase enzyme and restriction enzyme. Polymerase enzyme boolean many. Ligase, restriction enzyme. In the year 1960, 2 enzymes responsible for restricting the growth of bacteriophages in E. coli were isolated. Okay. One of this added methyl group to the DNA while the other cut DNA. This later was called restriction endonuclease. The first restriction endonuclease methyl group add करते हैं और पुलिस भी DNA लाइन काट देते हैं. The first restriction endonuclease is HIN2. First restriction enzyme endonuclease. 5 years later, it was found that HIN2 always cut DNA molecule at a particular 6 base pair. Specific sequence of 6 base pair. Recognition sequence for HIN2. 900 restriction enzymes that have been isolated from 230 strains, not 200, it's 230. 900 restriction enzyme from 230 strains. 230 strains of bacteria. Restriction enzyme 900. Okay, first letter genus, second two letters species, then or1 R strain, order of strain discovered. Okay. Eco RY13. Eco R1 is from Eco RY13. Y13. Eco RY13. The letter R is derived from the name of strain. Roman number following the name indicates the order in which the enzyme were isolated. Order in which the enzyme were isolated. RY13. Eco RY13. Eco RY13. 13. Of from the strain bacteria. Restriction enzyme belong to a large class of enzyme called nucleases. Restriction enzyme belong to a large class of enzyme called nucleases. These are two kinds, exonuclease and endonuclease. Restriction enzyme belongs to a large class of enzyme called nucleases. These are of two kinds, exonucleases and endonucleases. Exonucleases remove nucleotide from the end of the DNA, whereas endonucleases make cuts at specific position within the DNA. मुझे नहीं करना था ना. Exonuclease removes nucleotide from the end of the DNA, whereas endonuclease make cut at a specific position within the DNA. Nucleases. Enzymes called nucleases. These are of two kinds. Each restriction endonuclease functions by inspecting the length of DNA. Once it finds its specific recognition of a sequence, once it finds the specific recognition sequence, it will bind to the DNA. Once it finds the specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific. It will bind to the DNA and cut each of the two strand. of the double helix double helix at specific points in their sugar phosphate backbone. At specific points in their sugar phosphate backbone of the ferula, you will be fine. And cut each of the two strands of the double helix at specific points in their sugar phosphate backbone. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Position vector DNA, and this is genomic. Foreign DNA, eco-R1 cuts the DNA between the base G and A only when the sequence is GAA-TTC. Okay, once it finds a specific recognition, it binds to the DNA and cut each of the two strands of the double helix. Each. The nucleus recognizes a specific palindromic nucleotide sequence. Sticky ends. DNA fragments to insert sticky ends recombinant to DNA. Formation of recombinant DNA by action of restriction endonuclease. Enzyme eco-R1. 5' to 3', 3' to 5', GAA-TTC. GAA-TTC. Restriction enzymes cut the strand of DNA a little away from the center of the palindrome site. Restriction endonuclease cut the strand of DNA a little away from the center of the palindrome. Away from center of the palindrome. But between the same two bases on the opposite strand, this leaves single-stranded portion at the end. This leaves single-stranded portion at the end. They are named so beautifully. They are named so because they form hydrogen bonds with their complementary and complementary counterparts. This stickiness of the ends facilitate the action of enzyme DNA ligase. Restriction endonucleases are used in genetic engineering. To form recombinant molecule of the DNA. Restriction endonucleases are used in genetic engineering to form recombinant molecule of DNA, which are composed of DNA from different source genome. used in genetic engineering. Restriction endonucleases are used in genetic engineering to form recombinant molecule of DNA, which are composed of DNA from different sources or genomes. Composed of DNA from different source genome. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky end. And this can be joined together end to end using DNA ligase. Or in DNA, same restriction enzyme cutting both. Same restriction enzyme cutting both the DNA, foreign DNA and vector DNA at specific point. Same restriction enzyme cutting both foreign DNA and vector DNA at a specific point. Vector DNA plasmid. Same restriction enzyme cutting both foreign DNA and vector DNA at a specific point. Foreign DNA. This may not be true. Restriction endonucleases are used in genetic engineering to form recombinant molecule of DNA. Restriction endonucleases are used in genetic engineering. Restriction endonucleases are used in genetic engineering to form recombinant molecule of DNA, which are composed of DNA from different source genome. Restriction nucleases are used in genetic engineering to form recombinant molecule of DNA, which are composed of DNA from different sources genomes. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and this can be joined together end to end using DNA ligase. Joined together end to end. Foreign DNA, same restriction enzyme cutting both foreign DNA and vector DNA at a specific point. Ligase join foreign DNA to plasmid. Recombinant DNA molecule. Transformation, cell divide, E. coli. Cloning, host, E. coli, cells divide. Diagrammatic representation of recombinant DNA technology. Okay, separation and isolation of DNA fragment. The cutting of DNA by restriction nuclease. Source DNA with the same restriction enzyme. The recombinant vector molecule cannot be created. Recombinant vector cannot be created. Same restriction enzyme. The source DNA with the same restriction enzyme. Recombinant vector molecule cannot be created. Astaghfirullah, I can't do that. 372, something like that. Excuse me. Number 20. 60. Hey, I don't like it. What should I do? 270 plus 60. These fragments can be separated by techniques called separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease result in the fragments of the cutting of DNA by restriction endonuclease. The cutting of DNA by restriction endonuclease. Restriction endonuclease. The cutting of DNA by restriction endonuclease result in the fragment of DNA. The cutting of DNA by restriction endonuclease. Fragment of DNA. The fragment can be gel electrophoresis. Fragments can be separated by a technique known as gel electrophoresis. Fragment can be separated by a technique known as gel electrophoresis. Since these fragments can be separated by a technique known as... Why the heck? It's okay. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecule, they can be separated by forcing them to move towards the anode. Move towards anode, cathode. Cathode में negative charge, anode में positive charge. Largest to smallest. Well, are closest to cathode. Closer to cathode, smallest DNA. Closest to anode, largest. Okay. DNA bands. Cathode में positive, negative charge, positive charge on cathode. Negative anode, positive. Okay. A typical agarose gel electrophoresis showing migration of undigested lane 1 undigested and digested set of DNA fragment 2 to lane 2 to 4 digested undigested lane 1. Ethidium bromide staining with the DNA with the compound ethidium bromide followed by exposure to UV radiation. Exposure to UV radiation, you cannot see pure DNA fragment in the visible light. Bright orange. Okay, ethidium bromide, UV light, bright orange color. Elution. The separated band of DNA are cut out from the agarose cell. The separated band of DNA are cut out from the agarose cell. And extracted from gel piece. This step is known as elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with a cloning vector. Hi Adarsh. Ampicillin resistant, AMPR, tetracycline resistant, TET, tetracycline resistant. We have ORI, origin of replication, and here we have ROP. ROP is responsible for PBR32. Hind3, HIND, Hind3, Clav1, CLE, Clav1, and one more is there, Eco R1, Eco R1, okay, PVU1, PST1, we have BEMH1, and SAL1, origin of protein, PROTEN, protein responsible for cloning. Right? Protein involved in replication of plasmid. Protein involved in replication of plasmid. Protein involved in replication of plasmid. Replication of plasmid. Protein involved in replication of plasmid. Pust, Pervin pust, and here again we have PST, Pervin, PUV, P. हजुर ला केही भनेन त आन्टी मैले सोधेन पनि Hiya. Hiyo again. You're from Darjeeling. No, bro. I am from Sikkim, not Darjeeling. I think I'll come there after my exam, Darjeeling, with my friend and auntie. Bye. RUP, PUV2, RUP ma PUV2, okay, PVU1, PVU2, PVU2, RI. Origin of replication PBR 322 by H1, Sal H1, Lab 1, Hint 3. Protein replication. Replication of plasmid. Protein involved in replication of plasmid. PUC 8, PUC 8. Like an yeast artificial chromosome, bacterial artificial chromosome. Origin of replication. Replication start controlling the copy number of the linked DNA. High copy number. Origin pistol electron. So controlling the copy number of the linked DNA. Replication site. Replication start controlling the copy number of the linked DNA. Selectable marker. Alright. The vector requires a selectable marker which helps in identifying and eliminating non-transformant. Helps in identifying and eliminating non-transformant. And selectively omitting the growth of transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. You will study the process in subsequent section. Normally, the gene encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc., are considered useful selectable marker. Okay, ampicillin, tetracycline, or kanamycin, chloramphenicol are considered useful selectable marker. Are used as useful selectable marker for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc., are considered useful selectable tetracycline or kanamycin. Ampicillin, chloramphenicol, tetracycline, or kanamycin are considered useful selectable marker for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Okay. Ampicillin, chloramphenicol, tetracycline, or kanamycin are considered useful selectable marker. Okay. Ampicillin, tetracycline, kanamycin, chloramphenicol, etc. are useful selectable marker for E. coli. Recognition site. These are ampicillin genes encoding resistance to antibiotics. Resistance to antibiotics. Restriction site BAMH 1, SAL 1, HIND 3, CLAV 1, ECOR 1, PUV 1, PST 1, PUV 2. Okay. Praveen Past. Replication of the plasmid. DNA polymerase enzyme. DNA polymerase enzyme. Herbert Boyer. Stanley Cohen and Herbert Boyer. First, okay. Linking of gene encoding antibiotics resistance with the native plasmid. Y13 eco RY13 eco RY13 eco order in which enzymes were isolated. Chloramphenicol, tetramycin or kanamycin are considered useful selectable marker for E. coli. The normal E. coli do not carry resistance against any of these antibiotics. Cloning site. Preferably single recognition site. In order to link the alien DNA, the vector needs to have very few, preferably single recognition site for the commonly used restriction enzymes. Presence of more than one recognition site within the vector. Recognition site vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out at restriction site present in one of the two antibiotic resistance gene. to have very few, preferably single recognition site for the commonly used restriction enzyme. Codes of the protein involved in the replication of the plasmid. In order to link the alien DNA, the vector needs to have very few, preferably single recognition site for the commonly used restriction enzyme. In order to link the alien DNA, the vector needs to have very few, preferably single recognition site for the commonly used restriction enzyme. Alien DNA, the vector needs to have very few, preferably single recognition site for the commonly used restriction enzyme. उन्होंने नहीं दिखाया होगा ना. The vector needs to have very few, preferably single recognition site for the commonly used restriction enzyme. Presence of more than one recognition site within the vector will generate several fragments, which will complicate the gene cloning. Ligation of alien DNA is carried out at restriction site present in one of the two antibiotic resistance. The ligation of alien DNA is carried out at restriction site. Okay, cloning site and restriction site is same. Okay, the ligation of the alien DNA is carried out at a restriction site present in one of the two antibiotic resistance gene. For example, you can ligate a foreign DNA at BAM match 1 site of tetracycline resistant gene in the vector PBR322. The recombinant plasmid will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transferment on tetracycline containing medium. The transferment growing on ampicillin containing medium are then transferred. The transferment growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The transferment growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinant will grow in ampicillin containing medium, but not on that containing tetracycline, but non-recombinant will grow on medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transferment, whereas the other antibiotic resistance gene gets inactivated due to insertion of the alien DNA and helps in selection of recombinant DNA. Selection of recombinant transferment. Inactivated due to insertion helps in selection of recombinant. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure. PUC8. Blue-white screening. PUC8. Okay. PUC8. Alternative selectable marker. PUC8 have been developed which differentiate blue-white screening. Alternative selectable marker have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of a chromosemic substrate. In this, a recombinant DNA is inserted. Procedure color in the presence of chromosemic substrate. In this, a recombinant DNA is inserted within the coding sequence of the enzyme beta-galactosidase. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase. In this, a recombinant DNA is inserted within the coding sequence of the enzyme beta-galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. The presence of chromosemic substrate gives blue color. The presence of chromosemic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. The presence of chromosemic substrate give blue colored colonies if the plasmid in the bacteria. The presence of chromosemic substrate. The presence of chromosemic substrate gives blue colored colonies. The presence of chromosemic substrate gives gives the presence of chromosemic substrate gives blue color colonies if the plasmid in the bacteria does not have an insert. Presence of insert result into insertional inactivation of the beta-galactosidase gene and the colonies do not produce any color. These are identified as recombinant colonies. Vectors for cloning gene in plant and animals. Okay, ये भी पढ़ लेंगे, लेकिन क्या था तीन? Origin of replication, selectable marker, cloning site. Origin of replication, selectable marker, cloning site. And vectors for cloning gene in plants and animals. Okay, plants ma dicot ma हुन्छ हाम्रो Agrobacterium tumefaciens. Okay, animal ma retrovirus, bacteria ma था plasmid and bacteriophage. Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA, transfer DNA, tDNA, to transform normal plant cells into a tumor and direct this tumor cells to produce the chemical required by the pathogen. Similarly, retrovirus in animals have the ability to transform normal cell into cancerous cell. A better understanding. Okay, छोड़ दो ये. The tumor-inducing TI plasmid of Agrobacterium tumefaciens have now been modified into a cloning vector, which is now more pathogenic to the TI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector, which is no more pathogenic to the plants, but is still able to use the mechanisms. To deliver genes of our interest. There's no more pathogen, no, which is no more pathogenic to the plants, but still able to use the mechanism to deliver genes of our interest by into a variety of plant. Similarly, retrovirus have also been disarmed and are now used to deliver desirable genes into animal cells. Animal host, where it multiplies. Okay, competent host for transformation, recombinant DNA. Okay, ice, then 40 degrees Celsius heat, then again ice. Okay, बीच में क्या होता है? Ice, then incubation. Okay, DNA can be forced into such cells by incubating the cells with recombinant DNA on ice, followed by incubation of cell. First the incubation पर ऐसा था, मैंने गलत किया फिर. Incubating the cells with recombinant DNA. पहले ice नहीं था क्या? Treating. This is done by treating. तूने गलत बोला. Incubating the cells with recombinant DNA on ice. Incubating the cells with the recombinant DNA on ice, then 42 degrees Celsius heat shock, and back on ice. Treating a divalent cation, calcium. Incubating the cells with recombinant DNA. Incubating the cells with recombinant DNA on ice. Incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat shock, then putting them back on ice. Gun. Gene gun. Gold and tungsten. Coated with DNA. Gene coated. Okay, gold or tungsten. Coated with the DNA. Micro injection. Okay, animal's mouth. Directly injected into the number of animal cell. Micro injection. Animal cell. For plants, bombarded plants. Cells are bombarded with high velocity microparticles of gold or tungsten. Gold or tungsten. High velocity microparticles of gold or tungsten. Nucleus of an animal's. In other method suitable for plants, cells are bombarded with high velocity microparticles of gold or tungsten called the DNA in a method known as biolistic or gene gun. Gene gun. Gene gun, biolistic. 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 Gene gun, biolistic or gene gun. Gold and tungsten. Tungsten. Tungsten coated with DNA. The sound pathogen vector. Last method uses the sound pathogen vector, which when allowed to infect the cell, convey DNA into the host. Tungsten. Tungsten. 3 Process of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence पे मैं लिख देती हूँ ये. Process of recombinant DNA technology. Okay, recombinant DNA technology को. Processes of recombinant DNA technology. 1. Isolation of DNA. Fragmentation of DNA by restriction endonucleases. Isolation of desired DNA fragment. Ligation of DNA fragment into a vector. Transferring the recombinant DNA into the host. Culturing the host cell in a medium at large scale. And extraction of the desired product. Spooling. DNA that separates out can be removed by spooling. Chilled ethanol suspension. DNA involves several step sequence such as isolation of DNA, fragmentation of DNA by restriction endonuclease, isolation of desired DNA fragment, isolation of DNA, fragmentation of DNA by restriction endonuclease, Isolation of desired DNA fragment. Isolation of desired DNA fragment. Ligation of DNA fragment into a vector. Ligation of the DNA fragment into a vector. Huh? Isolation of desired DNA fragment. Ligation of DNA fragment into a vector. What the heck? Isolation of desired DNA fragment. Isolate करिए ना. Isolation of DNA. Fragmentation of DNA by restriction and nucleases. Isolation of desired DNA fragment. Recombinant DNA technology involves several steps in a specific sequence, such as isolation of DNA, fragmentation of DNA by restriction into nucleases, isolation of desired DNA fragment, ligation of DNA fragment into a vector. DNA fragment into a vector, ligation. Transferring the recombinant DNA into the host. Transferring the recombinant DNA into the host. Culture the host cell in the medium at large scale and extraction of the desired product. Culture the host cell in a large medium at large scale and extraction of the desired product. Isolation of genetic material. It's okay. Genetic material of all organisms, nucleic acid. DNA, deoxyribonucleic acid. DNA with restriction enzyme, it needs to be in pure form. It needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break it needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as RNA, proteins, polysaccharide, and also lipids. Since the DNA is enclosed within the membranes, we have to break the cell. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as RNA, protein, polysaccharides, and also lipids. Since the DNA is enclosed within the membranes, we have to break the cell open. Okay. Isolation of genetic material. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with the macromolecules such as, okay, we have to eliminate, okay, other macromolecules. DNA. We have to break the cell open to release DNA along with other macromolecules such as RNA, protein, polysaccharides, and lipids, lipases, ribonuclease, protein proteases, polysaccharides को लागि, chitin cellulose को लागि cellulase हुन्छ, lysozyme bacteria, the bacterial cell, peptidoglycan हुन्छ हैन. I'm not sure, but मलाई लाग्यो peptidoglycan नै हुन्छ. This can be achieved by treating the bacteria cell, plant cell, or animal cell with enzymes such as fungus को लागि chitinase, cellulase plant cell को लागि, and bacteria lysozyme, cellulase, and chitinase. Lysozyme, cellulase for plant, bacteria lysozyme, chitinase, fungus. You know that genes are located on long molecule of DNA intertwined with the protein such as histone. The RNA can be removed by treatment with ribonuclease, where protein can be removed by treatment with proteases. Other molecules can be removed by appropriate treatment, and purified DNA ultimately precipitates out after the addition of chilled ethanol. Other molecules can be removed by appropriate treatment and purified DNA. Other molecules can be removed by appropriate treatment and purified DNA. Chilled ethanol. Other molecules can be removed by appropriate treatment and purified DNA. Ultimately precipitates out after the addition of chilled ethanol. This can be seen as collection of fine threads in the suspension. The DNA that separates out can be removed by spooling. Cutting of DNA at a specific location. Restriction enzyme digestion. Cutting of DNA at a specific location. Restriction enzyme digestions are performed by incubating purified DNA molecule with restriction enzyme. The optimal condition for that specific enzyme, agarose gel electrophoresis, is employed to check the progression of restriction enzyme digestion. DNA is negatively charged molecule, hence it moves towards the positive anode. DNA is negatively charged molecule, hence it moves towards the positive electrode anode. This process is repeated with the vector DNA also. The joining of DNA involves several processes. Restriction enzymes, the recombinant vector molecule cannot be created. Why DNA requires separation through why DNA requires separation? Gel electrophoresis. Gel separation through gel electrophoresis. DNA separation through gel electrophoresis is necessary to analyze DNA fragments based on the size, allowing researchers to visualize and compare different DNA fragments, determine the size, and identify specific DNA sequence. Sieving effect. Agarose gel electrophoresis. DNA is negatively charged. The joining of DNA the joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA, with a specific restriction enzyme and cut out gene of interest from the source DNA and the cut vector with the space are mixed and ligases added. This result in the preparation of recombinant DNA, amplification of gene of interest using PCR. Amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of gene of interest is synthesized in vitro. Okay, vitro. Now, multiple copies of gene of interest using two sets of primer. A small chemically synthesized oligonucleotide that are complementary to the region of DNA and the enzyme DNA polymerase. The enzyme extends the primer using the nucleotide provided in the reaction and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times. That is, 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase, isolated from bacterium Thermus aquaticus, which remain active during the high temperature denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. 30 cycle, 1 billion, approximately 1 billion times. Amplified answer. Day. Denaturation, annealing and extension. Annealing must have been complete. Denaturation must have been basic heat. Denaturation. Double-stranded DNA, 5 prime to 3 prime, 3 prime to 5 prime heat. Reason to be amplified. Heat provided by annealing. Primer change must be present for further nucleotide addition. Starter is the काम गर्छ primer. Okay. DNA polymerase, stack polymerase, Thermus aquaticus polymerase. Okay. DNA polymerase plus deoxynucleotide. Tag polymerase plus deoxynucleotide extension amplified 1 billion times. 3 steps: denaturation, primer annealing, extension of the primer. Extension like synthesis extension of the primer. Okay. PCR stands for polymerase chain reaction. In this reaction, multiple copies of gene cloning. Gene of interest, multiple copies of gene of interest DNA synthesized in vitro using two primer. दुईटा प्राइमर ओके एउटा के हो ओलिगोन्युक्लियोटाइड अर्को एन्जाइम डिएनए पोलिमरेज र ओलिगोन्युक्लियोटाइड प्राइमर ओलिगोन्युक्लियोटाइड दैट इज डिओक्सिन्युक्लियोटाइड प्लस टैग पोलिमरेज जस्तै डिएनए पोलिमरेज ओलिगोन्युक्लियोटाइड टैग पोलिमरेज complementary to the region of DNA. The enzyme DNA polymerase. The enzyme extends the primer using the nucleotide provided in the region, that is the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of the DNA can be amplified to approximately billion times. 1 billion copies are made. Okay, DNA polymerase isolated from bacterium Thermus aquaticus. Insertion of recombinant DNA into the host cell organism. Ligated DNA into recipient cell. Recipient cell, after making them competent to receive, take up DNA present in the surrounding. So if the recombinant DNA bearing gene for resistance to an antibiotics, ampicillin, is transferred into E. coli cell, the host cell become transformed into ampicillin resistant cell. If we spread the transformed cell on agar plate containing ampicillin, only transformant will grow. Untransformed recipient will die. Since due to ampicillin resistant gene, one is able to select transformed cell in the presence of ampicillin. The ampicillin resistant gene in this case is selectable marker. Is called selectable marker. Okay. Obtaining the foreign gene product. Oligonucleotide, polynucleotide भन्दा पनि हुन्छ, deoxynucleotide भन्दा पनि हुन्छ, के को polynucleotide, tag polymerase, DNA polymerase, oligonucleotide, DNA, deoxynucleotide. Produce a desirable protein. Okay. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. Any protein encoding gene is expressed in heterologous host is called recombinant protein. Any protein encoding gene is expressed in a heterologous host. Any protein encoding gene is expressed in heterologous host is called a recombinant protein. Given it like recombinant protein. any protein encoding gene is expressed in a heterologous host. Any protein encoding gene is expressed in a heterologous host. It is called a recombinant protein. If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. The cell harboring cloned genes of interest may be grown on a small scale in the laboratory. If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. Let's see. If any protein encoding genes is expressed in heterologous host, it is called a recombinant protein. Recombinant protein. The cell can also be multiplied in continuous culture. Multiplied in continuous culture means a batch culture system where the used medium is drained out. Medium is drained out from one side, while fresh medium is added from the other to maintain the cell in their physiologically most active log exponential phase. Okay, first my log phase answer from the other to maintain the cell in their physiologically most active log or exponential phase. This type of culturing method produces a larger biomass, leading to higher yield of desired protein. Small volume culture cannot yield appreciable quantities of product. To produce in large quantities, the development of bioreactors, whereas 100 to 1000 liters development of bioreactors, large volume, 100 to 1000 liters of culture can be processed, was required. Astaghfirullah. Bioreactors can be thought of a vessel in which raw materials are biologically converted into specific product. Raw materials are biologically converted into specific products. Individual enzymes, etc., using microbial, plant, animals, or human cells. Bioreactors can be thought of vessels in which raw materials are biologically converted into specific products. Individual enzymes, etc. Basically, specific products and individual enzymes, etc., using microbial, plant, animals, or human cells. A bioreactor provides the optimal condition for achieving the desired product by providing optimum growth condition, temperature, pH, substrate, salt, vitamins, and oxygen. Stirring type bioreactors, most common stirring type bioreactors. Okay, एउटा simple हुन्छ एउटा के हुन्छ sparged, simple and sparged. Simple and sparged. Acid base for pH control, acid base for pH control, buffer. Steam for sterilization, sterilization. Steam for sterilization. Motor, foam breaker, flat bladed impaler, culture broth, nutrition, flat bladed impaler, foam breaker, motor, culture broth. Sterile air, increase surface area for oxygen transfer, gas entrainment, gas entrainment. Bubble dramatically increase the oxygen transfer area. Sparged more advantageous than simple. Curved to mix oxygen stirred. A stirred tank reactor is usually cylindrical with a curved base. Mixing of reactor content. Stirrer facilitates even mixing and Oxygen availability. Stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look, bioreactor has a agitator system, an oxygen delivery system, and a foam control system, a temperature control system, pH control system, and sampling port. So the small volume of culture can be withdrawn periodically. Downstreaming processes, purifying and preservatives, separation, purifying and processes, extraction of upstream process, biosynthetic stage. Before it is ready for marketing as finished product. Separation, purification, which are collectively referred to as downstream processing. Downstreaming processing. The product has to be formulated with suitable preservatives. Separation, purification process before it is ready for marketing as a finished product. The process includes separation and purification, which collectively refer to as downstreaming processes. The product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials, as in case of drugs. Strict qualities, strict quality control testing for each product is also required. Such formulation has to undergo through clinical trials, as in case of drugs. Strict quality control testing. Strict quality control testing. Strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. Such formulation has to undergo through clinical trials, as in case of drugs. Strict quality control testing for each product. Strict quality control. Strict quality control testing for each product is also required. The downstream processes and quality control testing vary from product to product. Plot. I don't know, something like that. Upstream process. Batch culture. Lag Z gene of interest, beta galactosidase. Colorless colony. G8 TC. I'll go and eat food, okay? टाइमर को पॉज करते हैं, 10 मिनट ब्रेक ले लेते हैं क्योंकि मुझे खाना खाना है. ब्रेक BREAK ब्रेक. Okay, biotech principle and processes. Okay. तो रिविजन इनफ नहीं था क्या? ये कल भी रिविजन करूँगी. केमिकल कोऑर्डिनेशन एंड इंटीग्रेशन एक बार फिर से. Okay. नभए त्यसरी भन्यो नि त आफू हेरेन तर भन्दा आफै हेरेको अनि आमाले के भन्यो म हो भने ए त्यस्तो हो कुरा चाहिँ तागेर नुफुमा च्वाचे हौ च्वाचे भन्दै मेरो राते त कति रमाउँदै खान्छ झन् त्यही त यहाँ वाट्सएपमा बोल्नु न तर यो कलमा त हुँदैन कि लाइभ भइरहेको छ लाइभ यहाँनिर चाहिँ बोल्नु ल अलरेडी मेसेज आइरहेको छ कि ओ होइन फिजिक्सवालाको त टाइममा बिटाइममा फिजिक्सवालाको के यहाँ छ नि त्यो एकछिनै बोल्नु एकछिन ल बोल्नु बोल्नु न रेकर्ड हुँदैछ अनलाइन रहेन रहेछ अफलाइन रहेछ यो भन्नु न
के गर्दैछ सर चोचे
भयो त्यो ए अनलाइनै रहेछ कि अस्ति कल गरेको थिएँ नि भन्नु त्यो भन्नु
अस्ति कल गरेको थिइस् नि किन गरेको
अब उसको टाइम सुन्यो एउटा हेऱ्यो उसले के भन्छ होला फोनबाट गर्न सक्दैन लाइभ सेसन स्टार्ट भएको छ भन्नु भिडियो कल गर्दैछ
फोन नगर है फोन नगर
एकछिन भएको छैन रेकर्ड यहाँबाट हुन्छ हौ एक्सेप्ट हुन्छ यो यताबाट पो हुँदैन त ल हाम्रो क्यामेरा अन हुँदैन जस्तो लाग्यो भयो त
के उर्मिला जस्तै देखेको
यो चाहिँ आमा होइन त हाँस्दाखेरि आमाको दाँतै एउटा देख्दैन के गर्ने हाँस्नु त के हो मातेर चाहिँ परि गएको जस्तै
के गर्दैछस् किन अम्ला टोप
कुनी
माथि रहेछस्
ए लुगा लगाएकै रहेनस् हौ
मातेर त बसेको त
अम्मा और दीना पीसी से ना बात कर रही है वीडियो कॉल पे ना, तो मैंने उनको वो दे दिया। अगर यहाँ पर रखती तो वो दोनों बात करते रहते ना, तो हम लोग डिस्ट्रैक्ट हो जाते, इसलिए दे दिया। त्यो आमा अलिकति पछाडि ठेल्नु त, पछाडि ठेल्यो भने आफै अन हुन्छ। स्क्रिनलाई पछाडि ठेल्नु न, मैले बन्द गरेको होइन, त्यो ल्यापटपै त्यस्तै छ हौ, आफै बन्द हुन्छ। मैना नमरिन्जेल। I need a pen. Thank you, Sri Ram Mishra. Oh, someone is there. Alana or something like that. मैं भूल गई फिर से तुम्हारा नाम. हेलो, इफ्तारी होगी यहाँ पर तो बहुत जल्दी होती है. चलो अभी बाय. ओके बाय अलाना. आई थिंक अलाना ही है. आई लिटरली फॉरगॉट योर नेम. After first piece, biotechnology principles and process part 2. हाँ. Molecular theory of inheritance. Nucleic acid. Nucleic acid is observed as a molecular unit of inheritance. Molecular theory of inheritance. Nucleic acid Nucleic acid is observed as a molecular unit of inheritance. Nucleic acid is observed as a molecular nucleic acid is observed as a molecular unit of inheritance. Molecular unit of inheritance. Gene. Discovery. First discovered as nuclein in pus cell RBC. Nuclein. First discovered as nuclein in pus cell RBC is observed as acidic substances, at the same time observed as phosphorus containing substances, observed by Mischer. Because of acidic nature, referred as nucleic acid by Altman. Nucleic acid is observed in polymer form. Nucleic acid polymer form. Nucleic acid is observed in two forms: ribonucleic acid, deoxyribonucleic acid. Ribonucleic acid. Ribonucleic acid. Ribonucleic acid. Common features: presence of nitrogenous bases. But one base is dissimilar in both. Common feature. Common features. Phosphate. Common features. Presence of phosphate. सी इज़ अननेसेसरी ब्लेमिंग में। मैं यहाँ पर बैठ के पढ़ रही हूँ ना, वो लोग दूसरे रूम में बात कर रहे हैं ना, बट स्टिल सी इज़ ब्लेमिंग कि मैं अम्मा को सिखा रही हूँ या फिर भड़का रही हूँ, इसलिए मुझे वो बिल्कुल पसंद नहीं है। मैं तो इन्वॉल्व हूँ लेकिन आई कैन हियर इट। गुस्सा आता है मैंने। न्यूक्लिक एसिड इज ऑब्जर्व्ड। बुरा भी लगा मुझे ये सब सुन के। Ribonucleic acid. Common feature. Ribonucleic acid. पीएफ निकालिस भन्नु न आफै। Ribonucleic acid. Common features: presence of nitrogenous base. Bye. Good night. दिनु। मैले सिकाउँदै थिएँ भन्दै थिइस् त, आमा त अर्को रुममा थियो त। मैले सिकाउँदै थिएँ आमालाई भन्दै थिइस् त, आमा त अर्को रुममा थियो, तेरो रुममा थियो त, मेरो कान छैन। निकाल दिया मैंने अपनी मन की भड़ास।
होइन सरी, मैले त्यस्तै सुने नि त।
के को छुट्टी?
भोलि जुम्मा गर्न जान्छ र सबै त पनि आज जुम्मा गर्छ भोलि।
भोलि फ्राइडे हो।
भोलि शुक्रबार।
कहाँ जान्छ? यहाँ मस्जिदै छैन। र गान्तोक जान्छ होला।
रानीपुरमा किन छैन?
कहाँ छ रानीपुर मलाई याद छैन
रानीपुरमा छ त त्यो उनीहरूको मल्टी छ त्यहाँ पैसा उठाउन आएको गर्छ अस्ति पैसा उठाउँदो रहेछ नि यिनीहरूलाई
ए अँ
कहाँ मुस्लिमहरू बस्छ त्यहाँ आउँदो रहेछ कि जाइनलाई त पैसै थिएन मसँग 50 रुपियाँ त थियो त्यही दियो भन्दै थियो सेतो टोपी लगाएर सेतो लुगा लगाएर हिँड्दो रहेछ मल्टी
मस्जिदमा त कहाँ जान्छौ केटीहरू
के थाहा हौ
होइन हौ आमाले तँ नि जाइ भने कि तँ नि प्रे गर भन्यो
प्ले गर्न घरमा
ए
आज रातभरि खा आज चाहिँ मोटले भन्दै थियो त्यत्रो दिन हुसुरिस एक दिन वर्ष बसिन भोलि के जुम्बा गर्छस् भनेको कि हाँस्छ मरेको आज त रातभरि खान्छु भोलि चाहिँ भोकै जुम्बा गर्नु हाँस्छ कुरा त ठिकै हो भन्छ 29 दिन कि 30 दिनमा चाहिँ एक दिन मात्रै वर्ष बस्दैछ अरे भोलि हो
ए अँ
अझै हाँस्छ पो तेह्र चाहिँ मलाई रिस उठ्छ कि गन गरिरहेको त गाली दिँदैछ
कसलाई ए उसलाई अरूलाई
अरू नेपाली नि त्यति बोल्नु जान्दैन कस्तो बोल्दैन फेरि गाली दिँदैछ मलाई एक्लै हाँस उठ्छ
दिँदैछु भन्नु नि त
अनि एउटा त्यो सालिक चाहिँ भन्छ कितलीले यहाँ चिया लिनु जान्छु भन्दो रहेछ कि ऊ चाहिँ काट्ला काट्ला भन्छ
के भनेको त्यो चाहिँ
कितली भनेको
ए अँ
काट्ला मोटुलाई के भन्छ को नि काट्ला काट्ला भन्छ
अनि हिन्दीबाट भनेको हुन्थ्यो त नि
त्यहाँदेखि काट्ला काट्ला भन्छ तेरो बाउको बाजेको काट्ला भन्छ झन् मरि हाँस्छ चिया बोक्ने भन्छ
ए अँ केटलीलाई चाहिँ कत्ला भनेको जस्तो सुन्यो होला नि
काट्ला नै भन्छ त्यसले कितली भन्नु जान्दैन खालि काट्ला
अनि उनीहरूले के भन्छ अरे
उनीहरूले पनि के भन्छ सोध्दिनँ त म अहिले त्यत्रो दिनमा चाहिँ भोलि जुम्बा गर्नु त
यो कस्तो पो छ त आमा त्यो मामा मिस्त्री छ नि उसलाई छोरा चाहिँदैसम्म चाहिँ डल्लै 30वटाको 30वटै रोजा बस्यो होइन बस्यो त छोरा आउँदा उसलाई चाहिँदाखेरि छोरा चाहिँदाखेरि उसको बुढी पनि बस्यो 30वटाको 30वटै बस्यो बुढा र बुढी नै
बसेन
नानी पाएपछि मात्रै बसेन
अनि उसले कहिले नि वर्ष बसेन पोहोर चाहिँ बुढीले कराएर बसेको अरे एक हप्तामा झन् मरेर चाहिँ भोकले मरेर चाहिँ त्यो छोड्यो त्यो बस्नै सक्दैन
nucleic acid okay molecular theory of inheritance or molecular basis of inheritance nucleic acid basis nucleic acid is observed as molecular unit of inheritance gene discovery first discovered as nuclein in pus cell RBC pus cell of RBC first discovered as nuclein is observed as acidic substance at the same time observed as phosphorus containing substance okay, observed by M. Misher because of acidic nature referred as nucleic acid by Altman. Nucleic acid is observed in polymer form. Nucleic acid is observed in two forms, ribonucleic acid and deoxyribonucleic acid. Ribonucleic acid or deoxyribonucleic acid. Common feature: presence of phosphate. Both are phosphate. Both are having pentose sugar, but in RNA has ribose. But RNA has ribose sugar and DNA deoxyribose sugar. Absence of oxygen at second position of sugar. Absence of oxygen at second carbon position of sugar. Common feature: presence of nitrogenous bases, but one base is dissimilar in both. RNA adenosine, guanosine, cytosine and uracil. DNA adenosine, guanosine, cytosine and thymine. RNA uracil, DNA thymine. Nitrogenous base purine. 9-membered double ring structure. Position of nitrogen is at 1379. Position of nitrogen at 13 and 79. Purine. 9-membered double ring structure. Purines, okay, nitrogenous base. Purine 9-member double ring structure. Position of nitrogen is at 1379. Pyrimidines, 6-membered single ring structure. Position of nitrogen at 1 and 3. Purine, adenine, 6th carbon of the ring, NH2 is present. Pyrimidine, okay, purine, adenine and guanine. Guanine, 2nd carbon, NH2. 6th carbon of the ring, NH2 is present. 6th carbon, double bond in guanine, C double bond, carbonyl group. Carbonyl group. Pyrimidines, pyrimidines, DNA, cytosine and thymine. 2 double bond 4 NH2. RNA, cytosine uracil, C double bond O at 2 and 4 of C in the ring, at 2 and 4 of the C carbon in the ring. 5th position, CH3 group is present, 5 methyl uracil. 5th position, CH3 group is present, methyl uracil, ribose sugar. Then ribose sugar and phosphate group, ribose sugar, phosphate group, P double bond O. Propose the component structure nucleotide in a polynucleotide chain. Base plus sugar nucleoside, base plus sugar and phosphate nucleotide, nucleoside plus phosphate nucleotide, base sugar plus phosphate nucleoside plus phosphate nucleotide, phosphate bond, glycosidic bond and glycosidic bond with purine. Component of nucleotide are connect. Components of nucleotide are connect to specific bonds. Respiration. Anatomy of flowering plant. Replication. 1,3 and 7,9 purine 1,3 position of nitrogen at 1,3 and 7,9 pyrimidine. Six-membered single ring structure. Position of nitrogen. 1379. Sixth carbon of the ring. H2 present. NH2 present on the sixth carbon of the ring. Sixth carbon of the ring. NH2 present. NH2 present on the sixth carbon of the ring. Guanine. NH2 second carbon. Sixth carbon. 0. Carbonyl group. Guanine. NH2 present on sixth carbon of the ring. Guanine. Second carbon. NH2 sixth carbon. Second carbon. Okay. NH2 present on in guanine. NH2 present on second carbon. Second carbon of the ring. Second carbon. Carbon of the ring. NH2 is present. Carbonyl group. Carbonyl present on sixth carbon. Carbonyl group. Carbonyl group. Sixth carbon, sixth carbon, NH2, sixth carbon में double bond of carbonyl group. Okay, sixth carbon of the ring, NH2 is present. Sixth, second carbon, NH2, second carbon, NH2, present on second carbon, present on second carbon, sixth carbon of the ring, NH2 is present. Sixth carbon of the ring, NH2 is present. Pyrimidine. Cytosine, thymine, cytosine, thymine. Thymine, cytosine, uracil. Second position. Cytosine. replication, CH3 group is CH3 group is present on 5th methyl uracil. CH3 group. CH3 group is present on 5th methyl uracil. CH3 group is present on 5th methyl uracil. Cytosine. Present on 2nd carbon, NH2, 4th carbon. Present on 5th methyl uracil. Present on 5th methyl uracil. 5th CH3 group is present on 5th methyl uracil. Double bond. 2/4 of carbon in the ring. 2 and 4th of carbon in the ring. Uracil. Double bond O. Double bond O is present on 2nd and 4th carbon of the ring. Of the carbon ring. Pyrimidine, purine, N on 1379. Pyrimidine 1 and 3. This is double ring. Purine, single ring, astaghfirullah, pyrimidine, astaghfirullah. Timing. Methyl, CH3 group on thymine, methyl group on 1st, 2nd, 3rd, 4th, 5th carbon. Methyl group on 5th carbon. Adenine, NH2 present on 6th carbon. 6th carbon. 1 in GUA. 1 in NH2 2nd carbon. NH2 2nd carbon. 6th carbon. NH2 2nd carbon. 1 in NH2 2nd carbon. 6th carbon. second carbon, sixth carbon. NH2 second carbon, sixth carbon. Second carbon, sixth carbon. NH2 second carbon हुन्छ, sixth carbon मा के हुन्छ? Guanine NH2 and second carbon. NH2 second carbon, sixth carbon मा C double bond हो. Carbonyl group guanine मा. Cytosine, uracil and thymine cytosine. Cytosine NH2 group कहाँनिर हुन्छ? NH2 on second position, fourth position, fourth carbon. Carbonyl group on second carbon, carbonyl group on second carbon. NH2 fourth carbon. CH3 methyl group is present on fifth methyl uracil. Uracil को double bond OS carbonyl group. Carbonyl group is present on second and fourth carbon in the room. दो बार carbonyl group. Fourth position में NH2. Second and fourth position में carbonyl group छ uracil को चाहिँ. Excuse me. Cytosine NH2 C double bond uracil second row fourth position मा छ thymine fifth CH3 methyl group present. Astaghfirullah. That was kind of toxic. Toxic treat. NH2 fourth carbon and double bond to second carbon. Second okay. To fourth adenine ma NH2 second sixth position cytosine guanine ma NH2 sixth ma double bond हो carbonyl group second ma, right? Second मा है गलत भयो. नहीं होगा शायद. cytosine. NH2 amine. NH2 amine, fourth carbon. Amine, fourth carbon. O oxygen, second carbon. C. Carbonyl. Cytosine. Go for C. To C. Cytosine. Uracil. 2,4. RNA, DNA. हाइड्रोजन छ भने सेकेन्ड पोजिसनमा हाइड्रोजन छ भने DNA, सेकेन्ड पोजिसनमा अक्सिजन छ भने RNA, सेकेन्ड पोजिसनमा अक्सिजन छ भने RNA. 2 3 4 5 1 2 3 4 5. Okay. CH2 OH. मेरो CH2 OH है ना, CH2 OH करेक्ट. तीन चार चारटा मात्रै छ, मैले पाँचवटा बनाएँ नि त. 1 2 3 4 5 त्यसो. CH2 OH. One, 2, 3, 4. 1 2 3 4. Okay, 1 2 3 4 5. Okay. Oxygen. OH. 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 OH3 तो सीधा से 1 2. Oxygen OXYZ. Second carbon no OH. Oxygen presence. Okay. RNA. This is RNA ribose sugar. Deoxyribose sugar ma चाहिँ same. CH2 OH. CH2 OH. OH. Hydrogen OH. This is DNA. Deoxyribose. second carbon na no oxygen. PO4 3 minus PO4 3 minus H3 PO4 3 minus phosphate group PO4 3 minus leaving. What's the component structure of nucleotide in a polynucleotide chain in a polynucleotide chain? Base plus sugar, nucleoside, base, sugar plus phosphate, nucleotide plus phosphate, nucleoside plus phosphate, nucleotide. Okay. Ribose sugar, deoxyribose sugar. Pentose sugar, one is deoxyribose sugar. Structure of deoxyribose sugar. CH5 CH5H10O4 1 2 3 4 5. Okay. Sugar, pentose sugar, PNTOSE, pentose sugar. One, two, three, 5. Second and fourth match है ना? Uracil. This is thymine. Structure of thymine, THYMIN, thymine. Thymine, structure of thymine. Pentose sugar. Ribose sugar. RNA. At second carbon. OH, CH2, OH. This is RNA. Okay. OH, CH2, OH, OH, OH माथि, OH तल, OH तल. This is RNA. DNA. 1 2 3 4 5 6 OH, OH, CH2, OH 3 4. Hydrogen. Phosphate group. phosphate. PO4 3 minus. PO4 3 minus. 3 minus. PO4 3 minus. Okay. O. H3PO4. H3PO4 ఉంటుంది. H3PO4 phosphate group. Structure. न्यूक्लिक एसिड या फिर न्यूक्लियोटाइड. बेस sugar and phosphate. Base sugar and phosphate nucleotide. Component of nucleotide are okay, thymine को structure I can make it, right? Thymine को रखियो यो pyrimidine हो. pyrimidine मा single ring हुन्छ. One ring. Nitrogen at 1 and 3 position. Nitrogen 1, 2, 1 6 membered हुन्छ हैन. अब त 6 membered 6 membered 1 2 3 1 2 3 2 1 2 3 कुन पोजिसनमा छ भनेको छ. यो चाहिँ थाइमिनको 2 र 4 मा कार्बोनिल ग्रुप. CH3 डबल बॉन्ड हो. CH3 छ. हो गया था ये थाइमिन था. Propose the component structure of nucleotide in a polynucleotide chain. Phosphate bond. Sugar र phosphate bond. Sugar र base बिचमा glycosidic bond. N-glycosidic bond with purine. First carbon sugar is attached to 9th position. Nitrogen. Pyrimidine C1 and 1. नलगाएको भने के हुन्छ नि केही नै भएको जतिकै अस्ति चाहिँ नि हाम्रो यहाँ अस्ति बेलुका हिजो बेलुका कि अस्ति बेलुका चाहिँ नि यहाँ यस्तो डर लाग्दो झुसुले यत्रो ठुलो अरू मात्रेको झुसुले चढ्दैछ कस्तो डर लागेको नि मार्नु मारे नि छोड्दा त झन् डर लाग्दैन कस्तो मार्नु कस्तो डर लाग्यो 5 मिनट जति म लगाएँ होला त्यति मार्नुको लागि आज चाहिँ हेरेको चाहिँ छुइनँ तर छर्नु चाहिँ हुनसक्छ हाई निधि गुड लक टू यू एज ओए वेरी शुभम आज वो आया ही नहीं इज दैट माय एलेक्स नहीं वो पहले का था यहाँ पर पॉज हो गया है गायब sure phosphate base phosphate bond between sugar and phosphate bond between sugar and nitrogenous base okay glycosidic bond and glycosidic bond with purine and pyrimidine okay and glycosidic bond C1N1 C1 sugar is attached to 9th position carbon 9 C9 okay nitrogenous base, nitrogenous base, phosphate sugar, phosphate sugar, phosphate, phosphate PHOS PHAT, phosphate PHOS PHAT, phosphate. Nitrogenous base. Okay. Phosphate. Nucleotide. Component of nucleotide. Component of nucleotide. Nucleotide. New component of nucleotide are connect with specific bonds. Sugar phosphate. Phosphate. This is phosphate bond. Nitrogenous base to sugar. Sugar. Base. And glycosidic GLYC glycosidic and glycosidic and glycosidic bond and glycosidic bond and glycosidic bond छ त्यहाँदेखि के छ phosphate छ यहाँनिर phosphate bond and glycosidic bond and phosphate bond. This is sugar and glycosidic bond, phosphate bond. And glycosidic bond. Purines, pyrimidines. Purines ma C1 and 1. C1 of sugar. C1 of sugar. Carbon 1 of sugar. And 9. And 9. C1 and 1. 1 3 ಅಂತ 1 3 nitrogenous base, carbon of sugar, nitrogen of nitrogenous base, of sugar को 1 फर्स्ट कार्बन सँग 9 N9 नाइट्रोजनस बेस को N9 अट्याच भएको हुन्छ के पिरिमिडिन्स मा C1N1 ओके C1 N9 र C1 respectively. Pentose sugar, ribose sugar, RNA, oxygen at second carbon. For ribose sugar, deoxyribose sugar, DNA, absence of oxygen at second carbon, DNA and RNA. DNA. Phosphate, PO4 3 minus, PO4 3 minus. Phosphate bond between sugar and salt. स्लॉट 3 पे कंप्लीट। यही हूँ। ओह अच्छा तुम यही हो। ये स्लॉट 3rd था ना, मैं शायद तब बाहर गई थी ना, तब मैं टाइमर ऑफ करना भूल गई थी। थोड़ा पढ़ लेते हैं ना, उसके बाद ही नेक्स्ट स्लॉट स्टार्ट करेंगे। एक्सक्यूज मी। ये कितना चालाक है शुभम। बॉन्ड्स, फॉस्फेट, नाइट्रोजनस बेस, शुगर and glycosidic bond. Okay, phosphate bond, sugar and phosphate, phosphate bond and glycosidic bond, sugar and base, between sugar and base. Okay, between sugar and base. Adenine, adenine, NH2, 6th carbon mount. Adenine, guanine, 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 NH2, 2 ma, 4 ma, cytosine, SINE, 6 ma, okay, 4 ma, 6 ma, right? No, cytosine, cytosine, double bond O, 4C, 2C2, carbonyl group 4 carbon मा हो। तो देखिए no 4C, no 4C, 2C, carbonyl group 2C, 4C मा NH2, no 4C2, C, cytosine, and second carbon NH2, second carbon NH2, cytosine को यो second carbon NH2, cytosine को तो NH2 4 carbon मा छ, करेक्ट, 2 carbon मा डबल बॉन्ड। ओके, cytosine यूरासिल इजी छ, 2 र 4 मा के छ, डबल बॉन्ड O छ। ओके, थाइमिन 5 मा CH3 हो नि त, राइबोस सुगर, डिओक्सिराइबोस सुगर, फस्फेट भयो, फस्फेट बन्ड हुन्छ नि, एन ग्लाइकोसिडिक बन्ड हुन्छ, एन ग्लाइकोसिडिक बन्ड प्युरिनमा सुगरको कार्बन बन्डसँग N9, पिरिमिडिनमा सुगरको बन्ड N1, नाइट्रोजनस बेस 1। Nucleoside and nucleotide of different bases, RNA. Adenine, guanine. Nucleoside, adenosine, guanosine. Nucleotide, adenosine, monophosphate, adenosine monophosphate. Nucleotide ma monophosphate. RNA, adenine, guanine. Nucleoside, adenosine, guanosine. Adenosine monophosphate, guanilic acid आइए तो होइन लाई यस्तो। Nucleoside, deoxyadenosine, deoxyguanosine. Nucleotide, deoxyadenosine monophosphate. एडिनोसिन मोनोफॉस्फेट दूसरे को तो लिखे ना रसा वानिलिक एसिड होला डीऑक्सीगुआनिलिक एसिड ऐसा तो नहीं होना चाहिए था डीऑक्सीगुआनोसिन साइटोसिन भाई ये तो लिखना पड़ेगा फिर मुझे साइटोसिन साइटोसाइटिडीन आरएनए साइटिडीन थाइमिडिन यूरासिल न्यूक्लियोसाइड गुआनिसिन पता नहीं क्या किया है कुछ तो किया है different bases RNA DNA adenine guanine cytosine thymine uracil uridine thymine 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 thymidine थाइमिन आरएनए में नहीं होता है ना ओके नॉट प्रेजेंट आरएनए यूरिडिलिक एसिड nucleoside and nucleotide of different bases $50, $60, $70, $85, $53, nucleoside and nucleotide of different pieces. Nucleoside and nucleotide of different pieces. Adenine, guanine, adenine, N-I-N-E, adenine, guanine, G-U-A-N-I-N-E, adenine, guanine, cytosine, thymine, uracil. Cytosine, S-I-N-E, cytosine, thymine, T-H-Y-M-I-N-E, thymine, and uracil. Uracil. Okay, adenine, guanine, cytosine, thymine, and uracil. RNA में ऐसे क्यों बना था? Nucleoside, adenosine, 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 deoxyadenosine, deoxyadenosine. Adenosine, deoxyadenosine, adenosine monophosphate, deoxyadenosine monophosphate. Okay, I don't think वहाँ पर होगा कि नहीं होगा, देखते हैं. Deoxyadenosine, R-E base. This is DNA. Adenosine and deoxyadenosine, these are nucleoside. Nucleoside. RNA, DNA, this is a nucleotide. Nucleotide. Adenosine, adenosine, deoxyadenosine, guanosine, adenosine, guanosine, G-U-A-N-O-S-I-N-E, guanosine, adenosine, guanosine, cytidine or cytosine, cytidine, D-I-N-E, cytidine, adenosine, guanosine, cytidine, थायमिन तो प्रेजेंट ही नहीं होता आरएनए में. यूरिडिन. Okay, adenosine, guanosine, nucleoside. Okay, these are nucleoside. Adenosine, guanosine, cytidine, uridine. Adenosine, guanosine, cytidine, and uridine. Nucleoside in DNA. Deoxyadenosine. Deoxyadenosine. deoxycytidine, deoxythymidine, deoxyadenosine, deoxy एडिनोसिन ही लिख दिया इसमें भी मैंने, पागल। डीऑक्सी एडिनोसिन, डीऑक्सी गुआनोसिन लिखना था ना इसमें। डीऑक्सी गुआनोसिन, डीऑक्सी गुआनोसिन, डीऑक्सी साइटिडिन, CYTI साइटी, डीऑक्सीसाइटोडिन, डीऑक्सीसाइटिडिन, डीऑक्सीसाइटिडिन, थाइमिन, डीऑक्सीथाइमिडिन, थाइमिन होता है क्या, थाइमिडिन, यूरिडिन, DNA में नहीं होता है। न्यूक्लियोटाइड, न्यूक्लियोटाइड इन RNA, एडिनोसिन मोनोफॉस्फेट, एडेनिन RNA में एडिनोसिन मोनोफॉस्फेट, PHOSPHAD, एडिनोसिन मोनोफॉस्फेट, डीऑक्सी एडिनोसिन मोनोफॉस्फेट, डीऑक्सी एडिनोसिन मोनोफॉस्फेट, फॉस्फेट, ओके। डीऑक्सी ग्वानिलिक एसिड, तो इसको एडिनाइलिक एसिड क्यों नहीं बोले? ग्वानिलिक एसिड, GUANYLIC, ग्वानिलिक एसिड, ग्वानिलिक एसिड, ग्वानिलिक एसिड, न्यूक्लियोटाइड, RNA में ना ग्वानिलिक एसिड, साइटिडिन, डीऑक्सीसाइटिडिन, साइटिडाइलिक एसिड, साइटिडाइलिक एसिड, एसिड, साइटिडाइलिक एसिड, थाइमिन नहीं होगा RNA में, यूरिडाइलिक एसिड, एसिड, थाइमिन ये नहीं होगा DNA में। डीऑक्सीसाइटिडाइलिक एसिड, डीऑक्सीथाइमिडाइलिक एसिड, डीऑक्सीसाइटिडाइलिक एसिड, डीऑक्सीसाइटिडाइलिक डीऑक्सीसाइटिडाइलिक एसिड, डीऑक्सीथाइमिडाइलिक, डीऑक्सीथाइमिडाइलिक, THOA थाइमिडाइलिक, 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 डाइलिक एसिड, deoxycytidilic acid, deoxythymidilic acid, deoxycytidilic acid, deoxythymidilic acid, deoxythymidine, deoxycytidine, deoxythymidine, deoxycytidine, cytidilic acid, uridylic acid, cytidilic acid, uridylic acid, guanilic acid, guanine, nucleotide of guanine, अच्छा छोड़ो यहाँ पर ही होगा। One in cytosine. Guanilic acid. कर दिया ना कांड। ग्वानिलिक एसिड, ग्वानिलिक एसिड डीऑक्सीराइबोस। इतना स्लो तो नहीं होगा। छोड़ो, नहीं है। Second position of carbon. After the oxygen is second carbon position of sugar. Nine-member DNA ring structure. Position of N at 79. 9-member double ring structure. Position of N is 1379. Purine 9-member double ring structure. Position of N is at 1379. हुआ तो nucleoside. It's already one. Already. Nucleoside, adenosine, guanosine, cytidine, uridine. Nucleotide. RNA. Adenosine. Okay. RNA को लगे adenosine monophosphate. Guanilic acid. Deoxyguanilic acid. Not sure. Okay. Deoxyguanilic acid. Deoxyguanilic acid. Adenosine monophosphate. Guanosine, deoxyguanosine, guanilic acid, deoxyguanilic acid. Cytidine, deoxycytidine, deoxycytidilic acid, deoxycytidilic acid. Thymine, छैन RNA मा के छैन. Deoxythymidine, RNA मा के छैन. Deoxythymidilic acid. Adenine guanine sites यहीं पर लिख सकते हैं. RNA में adenosine monophosphate AMP, adenosine monophosphate. GFP. Guanosine monophosphate is GMP. Guanilic acid is a nucleotide, a monomer used in RNA. तो भाई deoxyguanilic acid को बोलो ना. deoxyguanilic acid. कहाँ से नेट चल रहा है? वाईफाई तो चला गया तो. नहीं, वाईफाई ना फोन पे ना है, लेकिन ना पीसी पर कनेक्ट नहीं हो रहा है, पता नहीं क्यों. नो वाईफाई ऐसा कुछ दिखा रहा है. और नहीं, दो है ना, तो मैं 5G से कनेक्टेड हूँ. 2G वाला में नेटवर्क गया है और मैं पीसी पे ना 2G वाले से कनेक्ट थी. adenosine monophosphate, guanilic acid, cytidyllic acid, and uridylic acid. Cytidyllic, CITY, CITY, cytidyllic, CYTI. Cytidyllic acid, uridylic acid. न्यूक्लियोटाइड ही तो है ये ना, ये न्यूक्लियोटाइड ही है, न्यूक्लियोटाइड। ओके, डीएनए का होगा तुम्हारा डीऑक्सी AMP, एडिनोसिन मोनोफॉस्फेट, डीऑक्सी एडिनोसिन मोनोफॉस्फेट, डीऑक्सी गुआनिलिक एसिड, डीऑक्सी साइटिडाइलिक एसिड, डीऑक्सी थाइमिडाइलिक एसिड होगा, थाइमिडाइलिक एसिड, साइटिडीन, यूरिडीन, डीऑक्सी एडिनोसिन, डीऑक्सीगुआनोसिन, डीऑक्सीसाइटिडीन एंड डीऑक्सीथाइमिडीन। इतना ज़्यादा नहीं होता, आराम है भाई। डीऑक्सी लास्ट बोल, न्यूक्लियोसाइड में अपने न्यूक्लियोटाइड में डीऑक्सी एसिड, एडिनोसिन, डीऑक्सी एडिनोसिन मोनोफॉस्फेट, डीऑक्सी गुआनोसिन, डीऑक्सी गुआनिलिक एसिड, डीऑक्सीसाइटिडीन, डीऑक्सीसाइटिडाइलिक एसिड, डीऑक्सीसाइटिडाइलिक एसिड, डीऑक्सीथाइमिडीन, डीऑक्सीथाइमिडाइलिक एसिड, डीऑक्सीथाइमिडीन, साइटिडीन, साइटोसिन, साइटिडीन, गुआनोसिन, गुआनिन, एडेनिन, साइटोसिन, थाइमिन र यूरासिल, एडिनोसिन, गुआनोसिन, डीऑक्सी एडिनोसिन, डीऑक्सी गुआनोसिन, डीऑक्सीसाइटिडीन, डीऑक्सीथाइमिडीन। These are like almost 31 pages, not even half. DNA is a polynucleotide. Many nucleotide units. It started from dinucleotide. I think मुझे ना कॉफ़ी बना के पीना चाहिए। कब तक रहूँगी? सोचा था 3 तक रहूँगी, लेकिन नींद आ रहा है। कोई बात नहीं, मैं कॉफ़ी पी के आती हूँ। हाँ, कॉफ़ी पी के नहीं, ला के ही आऊँगी, इधर ही लेके आऊँगी। लेकिन हाँ निधि, सही बोला तुमने, 3 तक, अप्रॉक्सीमेटली 2:30 तक तो करेंगे। royal blue. Red, white and royal blue. 1,000 splendid sun. Dear stranger. Atomic habits. DNA is a polynucleotide. DNA is a polynucleotide, many nucleotide unit. Okay, polynucleotide. Oh yes, it starts from dinucleotide condition. Two phosphoester, one phosphodiester bond. Two phosphoester, one phosphodiester bond between two nucleotide. 3 end free, which hydroxyl end. Free hydroxyl end 3 prime, free hydroxyl group 5 prime, free phosphate 5 prime end, free phosphate. Phosphoester bond, phosphate bond, phosphoester bond. Okay, phosphoester bond, दो phosphoester bond, phosphodiester एउटाको 5 बाट अर्कोको 1 सँग ना 3 hydroxyl group छ नि त 3 free hydroxyl group and hydroxyl group 1 2 3 3 hydroxyl group 3 4 5 again 4 phosphodiester bond. Okay, in a single polynucleotide chain 4 bases. In a single polynucleotide chain ma 4 base, 4 bases are observed. What would be the total number of phosphodiester bond? 4 bases, एउटा दुईटा two phosphodiester bond, 3 लेखेको छ त यहाँ त. इन अ सिंगल polynucleotide 4 बेस, यहाँ एउटा बेस यहाँ छैन, अर्को बेस यहाँ छ, दुईटा भयो, यसको र अर्को तीनटा, दुईटा, एउटा दुईटा तीनटा, फेरि अझै एउटा छ भने. In a single polynucleotide chain 4 bases are observed. What would be the total number of phosphodiester bond and glycosidic bond? Okay, phosphodiester N-1, glycosidic bond N, where N is the base pair. जति बेस पेयर छ त्यति नै ग्लाइकोसिडिक बन्ड हुन्छ हो. N-1 हुन्छ phosphodiester bond चाहिँ. ओके, यहाँसम्म बुझेँ. phosphodiester 3, glycosidic bond 4, Pn-4 base, Pn. Polynucleotide chain 4 bases. Chain 4 base. So if base equals to N-1 phosphodiester bond, glycosidic bond N. DNA is the complementary. यहीं तक करो पहले. N-1 -1 is equals to number of phospho diester, phosphodiester bond. N minus 1 is equals to number of phosphodiester bond. N equals to glycosidic bond. Glycosidic bond. N minus 1 phosphodiester bond, N equals to glycosidic bond. Number of base. Nitrogenous base. Number of nitrogenous base present. Okay. N equals to number of nitrogenous base. N equals to number of base, nitrogenous base. N minus 1 number of phosphodiester bond. N equals to glycosid. N minus 1 number of phosphodiester bond हैन एउटा N minus 1 गर्छ. N equals to glycosidic bond हुन्छ. Number of base, nitrogenous base. N equals to number of base. N भनेको के हो? Nitrogenous base. Present in a polynucleotide chain. Polynucleotide chain हो जाएगा. क्या कर सकते हैं इम्प्रूव? एडिनोसिन दुईटा फर्मुला छ के फस्फेट बन्ड छैन. DNA is a polynucleotide. DNA is a polynucleotide. Many nucleotide unit. DNA is a polynucleotide. Deoxyribonucleic acid. All DNA polynucleotides होता नहीं. It starts from dinucleotide condition. 2 phosphoester, 1 phosphodiester. Okay. 2 phosphoester bond. 2 phosphoester bond. Is equals to 1 phosphodiester bond. 2 phosphoester is equals to 1 phosphodiester bond. Okay, phosphodiester bond. One between two nucleotide. दुईटा nucleotide को बीच में 5' carbon, 5' carbon. Phosphate group हुन्छ 5' carbon मा phosphate PO4 3-. 1 2 3. Okay, 5' carbon of sugar मा 5' carbon of sugar सँग 5' carbon free phosphate हुन्छ. Okay, 3 मा hydroxyl 3' मा free OH hydroxyl end 3' 3 hydroxyl end. Okay, फिर से एक बार इसको ना यहाँ पर बना के कर सकते हैं. 2 phosphoester bond is equals to 1 phosphodiester. ये phosphoester bond है ना. One, two, 3 4 phosphodiester बोला है ना इन लोगों ने. phosphodiester तो 1 2 3 4 5 6 होता. डाइएस्टर लिखा है. ये जो बॉन्ड दिखाया था इन लोगों ने फॉस्फेट बॉन्ड करके, ये फॉस्फेट phosphoester bond. Phosphoester. Phosphoester bond. Phosphoester bond. Okay. 5 prime मा phosphate हुन्छ, 3 prime मा hydroxyl group हुन्छ. Sugar को pentose sugar. Pentose मच्छर ना मैं तुझे मार डालूंगी. Pentose sugar. Pentose sugar. Free hydroxyl, 3 prime carbon. Free hydroxyl, 3 hydroxyl group. OH group, okay. OH group. Free hydroxyl group 3 prime माइना 3 prime of carbon मा hydroxyl group end. Okay. और कौन सा हाम्रो 5 prime carbon free phosphate, right? Free phosphate, PHOSPHAT, free phosphate group. Free phosphate group. PO4 3 minus group. PO4 3 minus. PO4 3 minus. Shillong. In a single polynucleotide chain, 4 bases are absorbed. What would be the total number of phosphodiester bond and glycosidic bond? Pentose sugar 3 prime carbon free hydroxyl group OH group 5 prime group free phosphate group PO4 3 minus PO4 3 minus group. Dinucleotide condition. In a single polynucleotide chain. Question in labor. Pentose sugar 3 prime carbon. Carbon of sugar 3 prime carbon of sugar 3 prime carbon. 3 prime carbon pentose sugar को free hydroxyl group OH group 5 prime carbon free phosphate group PO4 3 minus PO4 3 minus PO4 3 minus. Carbon PO4 3 minus. अस्ताको फेरुल्ला बेस यहाँनिर छ हाइड्रोजन एन्ड यहाँनिर के छ OH ग्रुप छ 3 प्राइम 1 2 3 3 प्राइम छ त्यहाँदेखि यहाँनिर के छ फेरि फस्फेट ग्रुप छ P oxygen, यहाँ के छ भन्नु त base. This is base 1, base 2. यहाँनिर कार्बन छैन न, कार्बन त्यता बिचतिर छ. CH2. फोस्फोरको अक्सिजनसँग आयो भनेपछि यो के हो? फोस्फोएस्टर. फोस्फोएस्टर, ESTer एस्टर बन्ड. फेरि यहाँनिर पनि के छ? कार्बन छ न, 3 प्राइम हो नि त, यो डाइरेक्ट OH सँग जोडेको छ. फोस्फोएस्टर बन्ड हो यो पनि. बेस 2 छ, यहाँनिर के छ? CH2 को युनिट छ, CH2. यो CH2 चाहिँ के छ? फस्फेटसँग जोडेको छ. एउटा हो अनि फाइनली एउटा हो. 5 प्राइम वाला एन्डमा के छ? फ्री फस्फेट ग्रुप छ. फ्री फस्फेट 5 प्राइम कार्बनमा के 3 प्राइम कार्बनमा यहाँ हेर त OH. फ्री 3 OH हाइड्रोक्सिल ग्रुप एन्डमा. बेस फस्फेट एउटा अक्सिजन यहाँनिर छ, दुईटा तीनटा के अक्सिजन छ. यो अक्सिजनमा चाहिँ कोसँग गरेको छ? CH2. 5th कार्बन हो नि सुगरको. यो अक्सिजन छ, BASE बेस. OH ग्रुप 3rd कार्बनमा. कार्बन हाइड्रोक्सिल ग्रुप 3rd प्राइममा के छ? हाइड्रोक्सिल ग्रुप छ. 3rd कार्बन अफ सुगर हो ल, यो कसको? सुगरको 3rd कार्बनमा फ्री हाइड्रोक्सिल ग्रुप छ. 5th मा को छ? फस्फेट ग्रुप छ. आएर जोडिन्छ. 3 प्राइममै त फस्फेट आएर जोडिनु पर्यो नि. ओके, यहाँ हेर त 3 प्राइममा चाहिँ 3 प्राइममा फस्फेट, 3 प्राइमसँग फस्फेट जोडिन्छ. 3 प्राइमको OH सँग फस्फेट आएर जोडिन्छ कि. भेरी इम्पोर्टेन्ट. दुईवटा फस्फोएस्टर बन्ड छ. फस्फोडाइस्टर 2 phosphoester and 1 phosphodiester. 1 phosphodiester. अस्ता पऱ्यो. 5 प्राइम कार्बन छोड्दैन हौ यीमा. 3 कार्बन हाइड्रोक्सिल ग्रुप on 3 prime carbon of any sugar, pentose sugar, SUGAR. It can let PO4 3 minus group attach to it. चारवटा बेस छ बेस जतिवटा छ माइनस n-1 फोस्फोडाइस्टर हो बेस जतिवटा छ त्यतिवटा नै के हुन्छ ग्लाइकोसिडिक बन्ड हुन्छ सुगर र बेसको बन्ड ग्लाइकोसिडिक बन्ड एन्ड ग्लाइकोसिडिक प्युरिन्स प्युरिनको 9 सँग हुन्छ n9 सँग C1 हैन सुगरको C1 n9 pyrimidine या फिर pyrimidine मा C1 n पनि 1 DNA is the complementary form of DNA is the complementary between the base. Okay, base AT 2 hydrogen bond, GC 3 hydrogen bond. Okay, complementary. DNA is the complementary form of anti-parallel strand of polynucleotide. DNA is a polynucleotide. यहीं पर लिख दो DNA. It is a polynucleotide. हाँ 10 बेस के बाद देख लेना एक बार इट इज अ पॉलीन्यूक्लियोटाइड दुईटा न्यूक्लियोटाइड एउटा न्यूक्लियोटाइड र अर्को न्यूक्लियोटाइड one phosphodiester between two nucleotide one phosphodiester it is a polynucleotide. It is the complementary form of anti-parallel strand of polynucleotide. Complementary form of anti-parallel. अब भाले त राति बाँच्दैछ त हाँ समझुको और त कसले कुखुरा पाल्छ र यहाँनिर 10 बजी पो बाँच्दै रहेछ के पो थियो complementary form of it is a polynucleotide complementary form of तो खुशी हो जाऊंगी ना तब तो DNA is a complementary form of अस्तगफिरुल्लाह मुझे ऐसा नहीं सोचना चाहिए DNA is a complementary form of anti-parallel strand of DNA it is a polynucleotide okay eta polynucleotide it is a polynucleotide DNA is complementary form of anti-parallel strand of polynucleotide it is a polynucleotide okay eta polynucleotide it is the complementary form of anti-parallel strand of polynucleotide complementary form of anti-parallel strand of polynucleotide छोड़ो कल की बात पुरानी it is a polynucleotide complementary form of anti-parallel strand of polynucleotide anti-parallel भनेको एउटा 5 प्राइम टु 3 प्राइम गएको छ नि अर्को के हुन्छ 3 प्राइम टु 5 प्राइम जान्छ this is anti-parallel okay it is the complementary form of anti-parallel strand of polynucleotide bases ATGC. GC. Three hydrogen bond. Bases. ATGC. Adenine र थाइमिनको बिचमा एडिनोसिन डिओक्सी एडिनोसिन र थाइमिन दुईटा एडिनोसिन र थाइमिनको बिचमा दुईटा हाइड्रोजन ग्वानिन डिओक्सी ग्वानोसिन साइटिडिन डिओक्सी साइटिडिनको बिचमा तीनटा हाइड्रोजन बन्ड ओके AUGC complementary with the DNA. RNA complements with the DNA. How many glycosidic bonds are present in DNA when the DNA contains 6 base pair? 6 वटा base pair छ भने नि base pair भनेको चाहिँ भनेपछि तीनटा base pair नि त एक base pair nitrogenous base base pair nitrogenous base इन्हें किसी और के साथ पेयर बनाएगा तो यहाँ पर भी होगा तो टोटली काउंट करो n equals to 6 एक में 6 वटा छ अर्कोमा पनि कति हुन्छ 6 6 into 2 12 12 bonds are present in DNA when DNA contains 6 base pair एउटामा एउटा दुईटा 6 base pair भनेपछि 6 वटा छैन 12 वटा हुन्छ टोटल 6 into 2 12 let me check how many glycosidic bonds are present in DNA when the DNA contains 6 base pair How many glycosidic bonds are present when DNA contains 6 base pair? Correct, 12. Para. तुम प्रधान इस क्वेश्चन को लिख लो How many glycosidic bonds are present? Present in DNA if there are 6 bases. Present in DNA when the DNA contains 6 base. When the DNA contains contains 6 base pair. How many glycans are present in DNA when the DNA contains 6 base pair? Okay. One strand. 6 base. One equals strand. There are 2 strands. And each strand has 6p. And there are 2 strands. Each strand has 6 nitrogenous bases. 6 into 12. Okay. Well, astaghfirullah. See, glycosidic bond. You will be fine, bro. You will be fine. Silent features of DNA. Complementary base pairing. Complementarily base pairing. Anti-parallel feature. Right hand turn coil. Watson and Crick model 1953. BDNA. Beta or BDNA represents certain angle between 2 base pair. Represents certain angle between 2 base pair. Okay. Right hand turn or coiling. Dexterous rotation. Each base pair rotates at a particular angle. Each turn equals to 360 degree. Each turn is equals to 360. In BDNA, in one turn, total 10 base pair are absorbed. क्या लिखा है ये मैंने? 360 degree 36 Armstrong. इतना कुछ लिखा है. This is just 5. This is page number 4. Silent features of DNA. Say silent features. Silent features of DNA. Complementarily base pairing. Complementary base pairing. Anti-parallel feature. Right hand turn coiling. Watson and Crick model 1953. Dextro rotation. Each base pair rotates at a particular angle. सबसे कंपेयर नहीं किया जाता है बहन. बीटा डीएनए या फिर बी डीएनए रिप्रेजेंट सर्टेन एंगल बिटवीन टू बेस पेयर. इट्स 360 के बारे में कुछ दिया है यहाँ पर. Watson, James Watson and Francis Crick. Watson and Crick, 1953. ओके यही तो है, लेकिन कहाँ दिया है? Now, it is made up of two polynucleotide chain where the backbone is constituted by sugar phosphate and base project inside. The backbone is 5 or something. It is made up of two polynucleotide chain where the backbone is constituted by sugar phosphate and the base project inside. The two chains have anti-parallel polarity. It means if one chain has the polarity 5 prime 3 prime, the other has 3 prime 5 3 prime to 5 prime. The base in two strands are paired through hydrogen bonds, H bond forming base pair. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with the cytosine with the 3 bond H bond. As a result, always a purine comes opposite to pyrimidine. This generates approximately uniform distance between two strands. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of the meter. That is 10 to the power minus 9 meter. Billion, okay. There are roughly 10 to the power 10 base pair. There are roughly 10 base pair in each turn. Consequently, the distance between the base pair in a helix is approximately equal to 0.34 nanometer. All I could say was Hajur. Silent features of DNA. Complementary base pairing. Anti-parallel features. Right hand. Okay. Complementary base pair. Anti-parallel feature. Features. Right hand turn. Right hand turn coiling. Hand coiling. Right hand. Dextro. Rotation. Dextro. Dextrous. Rotation. Dextrous rotation. Rotation. Watson and Crick model. model 1953. Each turn equals to 360. BDNA, okay, represents certain angle between two base pair. Okay, one turn. In one turn, there are total 10 base pair. ये भी के अंदर दिया है. 10 base pair in each turn. Consequently, base pair 0.34 nanometer. Total 10 base pair. 10 base pair. I have one turn. In one turn, there are roughly 10 base pair in each turn. There are 10 base pair. 10 base pair. Base pair. One turn. Astaghfirullah. One turn is equals to 10 pp base pair. So angle between two base pair is 36. The distance between base pair in a helix is approximately equal to 0.34 nanometer. Angle between two base pair. Angle between two base pair. Distance between. क्या लिखा है मैंने? Between two base pair 36 degree. 360 upon. ये नहीं लिखना पड़ेगा angle शायद. 360 upon. Base pair are present in one. 10 base pairs are present in one turn is equals to 34 Armstrong, 0.34 nanometer. So distance between two base pair is equals to 34 upon 10. That is 3.4 Armstrong, 0.34 nanometer. Okay, यही दो लिख देती हूँ मैं important point. एंगल को छोड़ दो. 36 डिग्री होता है एंगल. ओके, क्योंकि पूरा each turn equals to 360 डिग्री. एंगल पूछा है तो एक टर्न में 360, एक टर्न में 360. ओके, one turn 360 degree between two base pair 36 degree. The 10 base pair are present in one turn. The 10 base pairs are present in one turn is equals to 34 Armstrong, 0.34 nanometer. कोई लेना right? The pitch of a helix is 3.4 nanometer. Pitch of helix 3.4 nanometer. मतलब कि यही बोला है 3.4 nanometer. Pitch of helix. The 10 base pairs are present in one turn. 10 base pairs are present in one turn. In one turn there are 10 base pairs. Okay, one turn is equals to distance है ना. Pitch of helix यही लिखो. Pitch of helix लिखो. Pitch of the helix. PITCH. Pitch of the helix. Pitch of the helix is equals to, the pitch of the helix is equals to the base pair. The 10 base pairs are present in one turn is equals to 34 Armstrong. The 10 base pairs are present in one turn. 34. Pitch of helix 3.4 is equals to 3.4 nanometer or 34 Armstrong, right? Pitch of helix 3.4 nanometer. 1 meter is equals to 1 nanometer लिखना चाहिए. 1 मीटर है ना. 1 nanometer is 1 billionth of meter. 1 nanometer, nanometer, 1 nanometer equals to 10 to the power minus 9. 1 billionth meter, 1 nanometer, 1 nanometer ma 10 to the power minus 9 meter. Pitch of helix 3.4 nanometer or 3.34 Armstrong. Oh, the distance between two base pair. Distance between two base pair is equals to, first is nanometer ma 0.34 nanometer or 3.4 Armstrong. 3.4 Armstrong. 34 divided by 10. 10 16 था. 3.4 Armstrong. 0.34 nanometer. 0.34 nanometer. 1 nanometer is equals to 10 to the power minus 9 meter. Okay. Pitch of helix equals to 3.4 nanometer or 34 Armstrong. 3.4 nanometer. 10 base pair वकमा. 10 base pair 1 turn. Pitch of helix. PITCH. Pitch of helix. Okay. Complementary base pairing. Anti-parallel features. Right-handed coiling. Dexterous rotation. Watson and Crick model 1953. BDN. In 1 turn, pitch of helix, there are 10 base pair. 1 turn equals to 10 base pair. 1 turn is equals to 10 base pair. Pitch of helix is equals to 3.4 nanometer. 3.4 nanometer or 34 Armstrong. 1 nanometer equals to 10 to the power minus 9 meter. Pitch of the helix equals to 3.4 nanometer or 34 Armstrong. 0.34 nanometer or 3.4 Armstrong. Okay, if the somatic cell of human contains 6.6 into 10 to the power 9 base pair, what would be the length of DNA? 6.6. 6.6 into 10 to the power 9 base pair. Into 0.34 meter. Minus 9 meter. 66 को एउटा 0 आयो, दुईटा 0 आयो. 66 into 34 upon 1000. 66 into 34. 4 5 are 24, 6 are 24, 4 2 the way. 4 5 are 24, 6 are 24, 25, 26, 26, 3 6 are 18, 3 6 18, 19, 19, 4, 7 1 7, 7 is 14, 14 को 4, 1 इधर गया, 10, 11, 12, 1 इधर गया, 2 2 4 4 upon 1000, 1000 है ना, 2 1s are 2, 2 1s are 2, 2 2s are 4, 2 2s are 4 upon 2 5s are 10, 0 0, 2 5s are 10, इसमें 2 5s are 10 हुन्छ, 2 6s are 12, 2 1s are 2 upon 2 2s are 4, 2 5s are 10, 2 5s are 10, 2 55, तो 255 1s are 5 6 1, 255, 500, 250, 250, 500, 2 point something also, 2 point something, 555 na, 500, 250, 250, 500, 510 minus 1 5 0 51, 510. Okay, 2.2 meter, 510, 2.2 meter, 2.2 meter, correct. 6.6 into 10 to the power 9 base pair. मैं इसे छोड़ दो, इतने में ही छोड़ दो इसको. somatic cell of human contain diploid. Somatic means diploid, right? Diploid cell of human contains 6.6 into 10 to the power 9 base pair. The haploid cell of human 3.3 into 9 base pair. 0.34 nanometer. Okay, the length of DNA in E. coli is 1.36 meter. Base pair 4.6 into 10 to the power 6. Red cell of human. Pair in somatic. Somatic cell of human. If the somatic cell of human contains 6.6 into 10 to the power 9 base pair, what would be the length of DNA? 6.6 into. If the number of base pair in somatic diploid cell of human 6.6 into 10 to the power 9 base pair. Contains 6.6 into 10 to the power 9 base pair. Six point six into 10 to the power 9 base pair. What would be the length of a DNA? What would be the length of DNA? 6.6 into 10 to the power 9 into 0.34 Armstrong. Base pair, okay. ये एउटा भयो हैन त यसमा कति छ 10 to the power okay 0.34 3.3 into 10 to the power 9 base pair. 2n and haploid, okay. 6.6 into 10 to the power 9 base pair. 6.6 into 10 to the power 9 base pair. And that's okay. Got it. Base pair 6 into 10 to the power 9 base pair of 4.6 into 10 to the power 6 वास्तव फिर ला base pair BASE base pair 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 मुझे कुछ नहीं पता base pair of E. coli is equals to 4.6 into 10 to the power 6 1.36 meter 1.36 meter 1.36 meter 4.6 into 10 to the power 6 DNA is equals to 1.36. What would be the possibility of deviation in length when actual value is calculated? Length 4.6 into 10 to the power 6 into 0.34, 1.56 into 10 to the power 6. 4.6 into 10 to the power यो के हो भने त बेस पेयर हो 0.34 nanometer लेन्थ हो एउटाको दुईटा बेस पेयरको बीचको लेन्थ हो के 1.56 into 10 to the power 1.564 6. What would be the possibility of deviation in length when actual value is calculated? Total base pair तो पता नहीं क्या किया हैशटैग लगाइए देखते हैं क्या होगा डीएनए को लेन्थ ह्युमनमा कति हुन्छ 2.2 मिटर हुन्छ बेस पेयर डिप्लोइड सेलमा 2n मा कति हुन्छ 6.6 into 10 to the power 9 हुन्छ ह्याप्लोइडमा 3.3 into 10 to the power 9 हैन इकोलाइको लेन्थ हुन्छ इकोलाइको डीएनए को लेन्थ हुन्छ 1.36 मिटर 1.36 मिटर ह्याप्लोइड बेस पेयर कति छ र डिप्लोइड बेस पेयर कति छ डिप्लोइड 4.6 into 10 to the power 6 डिप्लोइड 4.6 into 10 to the power 6 भनेपछि ह्याप्लोइड कति हुन्छ 2.3 into 10 to the power 6 हुन्छ 4.6 into 6 okay 36 into 10 to the power meter on the base pair कति होला सन्दै हैन 0.34 nanometer 6 into 10 to the power 6 into 0.34 into the power minus 9 789 minus 3 4.6 into 0.34 into 10 to the power minus 3 एउटा दुईटा तीनटा 10 माथि आउँदाखेरि कति हुन्छ 46 into 34 प्लस माथि आउँदाखेरि माइनस कति हुन्छ 10 to the power minus 6 हुन्छ 6 into मिटरमै हुन्छ तर यो 6 into 34 4 5 are 24 6 24 को 4 2 यता गयो 4 4 जा 16 17 18 3 6 जा 18 को 8 1 यता गयो 3 4 जा 12 13 4 8 1 जा 8 8 16 16 को 6 1 यता गयो 3 4 5 okay 1.564 okay 3 हो नि त हैन 1564 into 10 to the power 6. और अभी 3 से गलत होंगे ऐसा stack features the sugar phosphate. The stacking features the sugar phosphate structure in the phosphonucleotide makes the main framework and the base and are present as inside branches from both polynucleotide strands. आ गए बीच में हैं nerve impulse conduction between neuron. Speed of conduction depends upon myelinated speed of myelinated action depolarization occur. Here, saltatory conduction, jumping conduction takes place. Speed of conduction depends upon diameter of axon. Large diameter directly proportional to speed of conduction. Threshold. Electrical. Stacking features the sugar phosphate structure. The sugar phosphate structure in the phosphonucleotide makes the main framework and तुझे नहीं बोला शुभम, मनास को बोला. मनासी है ना, वो पूछ रहा है ना कि कैसे पढ़े. तो उसे लग रहा है कि स्लॉट क्या होता है, इसे लग रहा है कि स्लॉट कुछ और है. कुछ और नहीं है स्लॉट. मुझे नहीं पता था 20 डिग्री सेल्सियस में भी ठंडा फील होता है, या फिर ये मेरी एरिया का नहीं है. 20 डिग्री सेल्सियस देखो तो एक बार. कहाँ गया, अभी तो यहीं था. ये मेरा ही है. 20 डिग्री है फिर भी ठंड लग रहा है. Constant Stacking features. The sugar phosphate structure in the sugar phosphate structure in the phosphonucleotide. Sugar phosphate structure in the phosphonucleotide makes the main framework, and the base are present as inside branch from both polynucleotide strands. Stacking feature. Stacking feature. The sugar phosphate structure in phosphonucleotide makes the main framework. Main framework. Main framework. Sugar phosphate structure in the phosphonucleotide makes the main framework, and the bases are present as inside branch from both polynucleotide strand. Main framework. Sugar phosphate structure. The sugar phosphate structure in the phosphonucleotide makes the main framework. Sugar phosphate structure in the phosphonucleotide. Main framework. Sugar phosphate. Sugar phosphate structure. Sugar phosphate structure PHOS. Sugar phosphate structure. The main framework. Sugar phosphate structure. Okay, in phosphonucleotide the sugar phosphate structure in the phosphonucleotide makes the main framework, and bases are present as inside branch from both nucleotide strand. The sugar phosphate structure in the phosphonucleotide phosphonucleotide makes the main framework, and the bases are present inside branch strand bases inside the branch. Bases. In bDNA, the bases are present in one plane stacking over each other. In bDNA, the bases are present in one plane stacking over each other in a polynucleotide. Bases are present in one plane stacking over each other. Nucleotide TID, nucleotide chain. Astaghfirullah. Main framework, sugar phosphate structure, inside branch, bases. In bDNA, bases are in one plane stacking over one another. In bDNA, bases are in one plane stacking over each other in a polynucleotide chain. Chargaff's rule, proposed by E. Chargaff. Proposal was based on complementary base pairing. टाइटल हो बेबी रोम नहीं है, ठीक है। पेज नंबर 5 यहाँ पर लिखो, पेज नंबर 5, पेज नंबर 6। Proposed by Chargaff. Proposal based on complementarity. Based on complementarity. Based on complementarity. Base pairing. Base pairing. Based on complementarity, postulate sum of purine. Postulate sum of purine is equals to sum of pyridine. Pyrimidine. Postulates. T plus Z, ATGC. G1 the ratio. Purines A plus C. The ratio A plus C plus G upon the ratio purines upon pyrimidines is equals to 1. C plus D, A plus D plus C plus A plus G plus A plus G upon C plus D is equals to constant 1. Equals for all species 1 plus G. Purine, purine upon pyrimidine. Pyri is equals to 1, constant for all the species. And for all species, purines A plus G upon T plus C. Adenine plus guanine upon cytosine plus thymine is equals to 1, constant for all species, okay. 4A, 4G, T4, C4, 4 plus 4, 4 plus 4, 4G, C4. The ratio of A plus T upon G plus C, less than 1, more than 1, okay. Less than 1 prokaryotic condition, more than 1 eukaryotic condition. Specific value different from different species, value different for different species, but for one species, but for one species K, specific value different for different species. भाई साहब ये क्या दिया है, मुझे तो समझ में भी नहीं आया. This is so irritating, bro. क्या करें? A plus G upon C plus D equals to 1. Constant for all species. The ratio of A plus D the ratio of A plus D upon B plus C is equals to specific value for different species. Prokaryotes and eukaryotes. A plus D upon G plus C less than 1, more than 1. Eukaryotes more than 1. Less than 1. Greater than 1. Excuse me. Okay, I don't know. percentage of A in the percent A in DNA is 40%. 40% A. Then what would be the percentage of D? 40%. C 30%. 40 40 80. C 20% and G 20%. C and G 20%. 40 to 40. Sixty, 80, 90, 100 plus 40, 80. Percent. 90, 100. DNA as a genetic material. Sorts of DNA as genetic material followed by 3 sets of experiment. Griffith's transforming principle experiment. Avery, McCarty and McLeod. Biochemical nature of transforming principle and Hershey-Chase experiment using radioisotope P22 and S35. Phosphorus, P phosphorus. P phosphorus S16 15 22 का नहीं हो जाएगी P22 and S35. Confirm DNA as Hershey-Chase. Confirm DNA as genetic material. DNA as genetic material. Material, okay. Search. Griffith transforming principle. Biochemical nature of transforming principle. Biochemical nature of transforming principle. Biochemical nature of transforming principle. Griffith transforming principle. Nature of principle. Biochemical nature of transforming principle. Hershey-Chase. Hershey-Chase experiment using radioisotope. Hershey. Hershey and Chase experiment. Chase experiment. Excuse me. Using isotopes, radioisotopes, okay. Confirm DNA as genetic material. Hershey-Chase experiment. Radioisotopes, TOPS, isotopes P22 and S35. Sulfur 35. 22 and has 35. Okay, radioisotopes. Hershey and Chase experiment हो confirm. Okay, Hershey and Chase लेने की करी confirm. DNA is unequivocal proof of DNA as genetic material. Unequivocal proof of DNA is what? Unequivocal proof of DNA as genetic material. Griffith experiment. Salmonella. No, Streptococcus pneumonia. Griffith GRIF. Griffith's experiment. (1982) 1928. Two strains of bacteria were taken, R strain and S strain. Streptococcus pneumonia, previously referred to as Diplococcus or pneumonococcus. ये तो नहीं आएगा. Streptococcus pneumonia, test organism. Streptococcus pneumonia. Streptococcus coccus. Streptococcus pneumonia. PNEU. Pneumonia. Streptococcus pneumonia. Test organism. Streptococcus pneumonia. Bacteria. ये उठा क्यों? Bacteria. Two strains. Two strains. R strain, absence of mucus layer, non-virulent. R strain, 2R, 3S. Two strain. R strain, STRAIN, R strain, 2R. S strain, STRAIN strain, 3S. Two strains, okay. Streptococcus pneumonia. Test organism Streptococcus pneumonia. Test organism Streptococcus pneumonia. अच्छा. R strain and S strain. R strain म्हणजे एउटा S strain हुन्छ. R strain. Absence of mucus layer, non-virulent. Smooth layer of mucus due to polysaccharide. Virulent. Okay, S strain harmful रहेछ. Hershey and Chase experiment, packaging of 5 ये भी करके ये ग्रीन वाला पोर्शन पूरा कंप्लीट करो, उसके बाद देखेंगे आगे का. Griffith experiment 1928. Test organism Streptococcus pneumonia bacteria. 2 strain, R strain and S strain. R strain. Absence of mucus layer. SANC. Absence of mucus layer. MUCS. Mucus layer. Absence of mucus layer. Non-virulent. Absence of mucus layer. Non-virulent. R strain, 2R, absence of mucus layer, non-virulent, whereas S strain, presence of mucus due to polysaccharide. Smooth layer, SMOOTH, smooth layer due to presence of polysaccharide, SACCHARID, polysaccharide. Smooth layer because due to polysaccharide they are virulent. Smooth strain wala virulence, okay. Harmful. Experiment. R strain alive plus mice. Mice leaves. S strain heat killed plus mice. Mice leaves. R strain live plus mice. Mice dies. R strain live. S strain heat killed. Dies. Okay. Chatta recess experiment. Experiment of Griffith. GRIITH. Griffith. R live. R strain alive. R strain plus mice bani alive hai. Mice lives lives 2 number मा छ R strain अस्तक फेरल्ला S strain heat killed plus mice leaves. S strain R strain heat kill plus mice. Dies. R strain live. S strain heat killed. Mice dies. Okay. R strain is transformed because of virulent S strain. Transforming principle. R strain change. R strain is transformed because of virulence of S strain. Conclusion. R strain, STRAN strain, back is transformed. TRANSFORMED, transformed by because of virulence of S strain. Transformed because the S strain and Griffith, GRIFFITH, Griffith referred this change. REFFERRED referred this transformation. FORMATION, transformation as transforming principle. Transforming, forming principle. R strain bacteria is transformed because of the virulent gene of S strain. R strain bacteria is transformed because of the virulent gene of S strain. And Griffith referred this transformation as transforming principle. R strain bacteria is transformed because of the virulent gene of S strain. And okay. And Griffith referred this transformation as transforming principle. Biochemical nature of transforming principle. Avery, MacLeod and McCarty. वहाँ पर तो मैंने लिखा भी नहीं. वाह भाई वाह. देख लेंगे. क्या देख लिया? Biochemical nature of transforming principle. Astaghfirullah. By Avery, MacLeod and McCarty. MacLeod. McCarty. By Avery, MacLeod and McCarty. Biochemical nature of transforming principle. Experiment. Isolation of 3 enzymes. Protease, DNAs and RNAs. Breakdown of denature the protein, denature RNA, RNAs, denature of DNA. Isolation of 3 enzymes. Isolation of 3 enzymes. Protease, protease, RNAs, DNAs. Same set of Griffith experiment was used. Live R strain plus heat kill S strain. Transformation. Culture mixture. 1 by 1 enzyme checked by introducing in the culture mixture. R strain, heat kill S strain plus protease. Transformation. Absorbed. Same set of Griffith experiment used. Same. Used. First. Strain. Strain alive, live. Oh. R strain plus heat kill S strained. heat kill. Astaghfirullah. I'll die. Protease. 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 Transformation. Observe no role of protein. Observed. No. Protein. Transformation. Concluded. Transformation. R restrain, heat kill S restrain, RNA is transformation observed. RNA is not transforming principle. Same conclusion. DNAs, no transformation. R strain, heat kill strain. DNAs, no transformation. DNA is the biochemical nature of transformation. No transformation. TRANS RMATN. Transformation. No transformation. Therefore, DNA is the biochemical nature of transformation. DNA was the biochemical. The DNA was the biochemical nature of transformation. Transformation. Third. Still not concluded about DNA as genetic material. Concluded about the DNA as genetic material. Could not consider DNA as genetic material. But still could not consider DNA as genetic material. Hershey and Chase experiment. Hershey and Chase experiment. Okay. Unequivocal observation of DNA as a genetic material by using radioisotope method. एक phosphorus 32 था, मैंने 22 लिख दिया भाई साहब, शर्म कर बहन. 32 35 S35 है ना, P32 and S35 यहीं पर भाग रही है एग्जाम. P32 and S35. Oh. Observation of DNA as genetic. Observation of DNA as genetic material by using radioisotopes method. P32 as a component of DNA. S35 as a component of protein using radioisotopes method. P32 phosphorus 15 है ना, as a component of DNA. Phosphorus 32 as a component of DNA. S32 नहीं, S35 as a component of protein. S35 as a component of protein. Includes the T2 bacteriophage DNA protein. Undergoes lytic cycle for reproduction. Use E. coli bacteria. Include T bacteriophage. What is T2 bacteriophage? T2 bacteriophage. Alfred Hershey and Martha Chase. Bacteriophage. Infect bacteria. Bacteria E. coli. Bacteriophage. T2 bacteriophage. DNA and protein पनि छ हो. Undergoes lytic cycle for reproduction. Use E. coli bacteria. Experiment. Phosphorus 32 एक में होगा okay. Phosphorus 32 S35 P32 infection. P32 S35 radioisotopes. No radioisotopes. Phosphorus. Infection blending to separate the two entities. Protein remains outside of the bacteria. S35, okay. Centrifugation as supernatant. Radioisotope P32. Bacterial mass. Palette. S35 radioisotopes. virus supernatant S35 radioisotopes, bacterial mass palette radioisotopes P32. P32 initially belonged to genetic material of virus, passed to bacteria because palette of bacteria contains radioisotopes is part of DNA. Radioisotopes contain palette of bacteria contains radioisotope which is a part of DNA. DNA is a genetic material. बस इतना ही. फिर कभी infection, blending and centrifugation. 3 steps include T bacteriophage, T2 bacteriophage, include bacteriophage, PHEG, virus that causes disease, virus that causes disease in bacteria. That infect bacteria. Bacteria E. coli. Okay. DNA protein. 3 steps. Infection, 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 centrifugation and infection, blending and centrifugation, IBC, infection, blending and centrifugation, 1, 2, 3, centrifugation, infection, blending and centrifugation. Bacteriophage, radioactive S35 labeled protein capsule, radioactive P32, 35 labeled protein capsule, radioactive P32 labeled DNA, infection. Infection by blending, centrifugation. No radioactive S35 detected in cell. S35 detected in supernatant. Radioactive P32 detected in cell. No radioactivity detected in supernatant. The Hershey-Chase experiment again. Suspension. अरे ब्रो, स्लॉट 5 क्या है ये पूछ रहा है. वहाँ पर लिखा है तो एक स्लॉट में 50 मिनट होता है, 10 मिनट का ब्रेक लेते हैं, लेकिन अभी ब्रेक नहीं ले रहे क्योंकि एग्ज़ाम पास से स्ट्रेस ज़्यादा है. हाँ, सारे 2 तक पढ़ेंगे. पढ़ो ना. हो तुम लोग क्या बकवास की क्वेश्चन पूछ रहे हो. प्लीज़ आदित्य पढ़ो चुपचाप बैठ के. मैंने आदित्य को पहले भी देखा था शायद। चलो क्या करना, ये एग्जाम में नहीं आएगा। मेरा कि वो डिपेंड करेगा कितने बजे उठेंगे। ऐसा सच कोई टाइमिंग नहीं है। मैं ना चैट्स ऑफ ही कर देती हूँ, तुम लोग मेरी बात सुनने तो वाले नहीं हो। भाड़ में जाए, अस्ताफिरुल्लाह नहीं बोलना चाहिए। 50 18.3। Infection blending, 3 steps on infection blending, centrifugation, centrifugation. DNA protein. Protein. Undergoes lytic cycle for reproduction. Use E. coli bacteria. Experiment. Hershey-Chase. S35. P32. Radioactive DNA. S35. Radioactive protein. Radioactive protein. Radioactive protein. PROTEI. Radioactive protein. Okay, S35. Pure radioactive protein. Radioactive DNA. Radioactive protein. Infection. First step. Infection. Infection. أستغفر الله. S35. Blending. P32. P32. Blending. This is E. coli. This is another E. coli. भाई साहब मुझसे नहीं होगा इतनी कसरत. Protein remains outside the bacteria. P32. Protein remain outside the cell. S35. Okay, bahar. P32 inside. Blending to separate the two entities. Blending. to separate. separate, separate the two entities. Third step, centrifugation. Third step. Centrifugation. 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 क्या था इसमें? Virus. Supernatant. S35. Supernatant. बैक्टीरियल मास. क्या क्या दिखाया है? कुछ नहीं दिखाया है. Palette. Thirty-five. Radio isotope B32. BACTERIAL. Bacterial mass. Massless palette. PELLET palette. Okay. पता नहीं ये क्या है. Supernatant. 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 Isn't. Initially belong to P32. P32 initially belong to genetic material of virus. P32 initially belonged to genetic material of virus, passed to bacteria. Or bacterial mass. P32 virus, but passed on to virus was passed, PASSED, passed to bacteria. Because pellet of bacteria contain radioisotopes. Bacteria. The virus was passed to bacteria as the pellet, pellet of bacteria contain radioisotope. DNA. Material. हो गया. Twinkle, twinkle, little star, how I wonder what you are. All above the world so high. So gross. Bloom. आलसी इंसान. Packaging of DNA in chromosome. DNA include many base pair. In humans, 6.6 into 10 to the power 9 base pair. In diploid, 3.3 into 10 to the power 9 base pair. In haploid, E. coli, 4.6 into 10 to the power 6 base pair. In human, length of DNA 2.2. In E. coli, 1.36. This represents condensation and packaging in small length chromosome. This represents condensation and packaging in a small length chromosome. Chromosome is the functional gene, functional DNA, okay? Chromosome is the carrier, functional gene. Chromosome carrier, functional gene. Condensation packaging includes histone protein. Basic nature due to lysine and arginine. Consists of 5 histone units: H1, H2, H2B, H3, H4. These 4 are present in, what is this? Duplet form twice. Different nucleosomes are connected with each other through linker DNA small fragment. H1, H2A, H2B, H3 and H4. H1, H2A, H2B, H1, H2A, H2B, H3 and H4. 5 H1. These 4 are present in duplet form twice. 2 into 4, 8. Octamer complex. देखते हैं क्या होता है. आगे बढ़ो लेडी के प्रकार. यहाँ पहला हेल्पिंग 6 7 8 9 10. Packaging of DNA in chromosome. ये ऊपर क्यों मुड़ गया? These represent condensation and packaging in small lengths. Chromosome, functional gene, carrier functional gene, functional DNA. Condensation and packaging. Carrier functional gene. Chromosomes carrier functional gene. Condensation and packaging of DNA in small length. Packaging of DNA in small length. Functional DNA. Condensation and packaging of DNA in small length. Carrier functional gene. Functional gene. Functional DNA. Condensation and packaging includes histone protein. Condensation and packaging. Condensation and packaging include histone protein. Condensation and packaging include histone protein. अरे भाई साहब क्या? Condensation packaging includes histone protein. Histone consists of 5 histone units. Histone protein. Basic due to lysine and arginine. C-O-N-E, histone. Basic. Lysine and arginine, histone, arginine, N-I-N-E, arginine. Basic protein, lysine and arginine. Histone protein, P-R-O-T-E-I-N, protein. Basic protein due to lysine and arginine. Lysine and arginine consist of 5 histone units. Consist of 5 histone unit. Consist of 5 histone unit. H-I-S-T-O-N, histone unit. U-N-I-T, unit. Consist of 5 histone unit. H1, H2A, H1, H2A, H2B, H3N, H4, H2A, H2B, histone H1, H2A, H2B, H3N, H4. H2A, H2B, and H3 and H4. These four are present in diplet form. These four are present in duplet. D-U-P-L-E-T, duplet form. Duplet form, okay. 24. Octamer complex. OCTA, octamer complex. 4 into 2 is equals to 8. तीन चार पाँच छः सात आठ H2A, H2B, H3 and H4. Octamer complex. Thirteen upon 4. Turn 1/4 below. 3 upon 4. Octamer. Stone 1, H1. Contain around 200 base pair of DNA. 200 base pair. Base pair. Nitrogenous base pair. Different nucleosomes are connected with each other through linker DNA. Small fragment. Beads on string appearance converts to solenoid appearance. Diameter 10 nanometer. 1 octamer. Both the amino acid residues carries positive charge in their side chain. Stones are organized to form a unit of 8 molecule called histone octamer. The negative charged DNA is wrapped around the positive charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pair of DNA helix. Histone DNA. Nucleosome. 3/4 turn. Octamer. Okay, nucleosome. Nucleosome. S-O-M-E. Nucleosome. H1 histone. Octamer. One 3/4 turn. 3/4 turn. Contain around 200 base pair of DNA. Nucleosome. 200 base pair of DNA. Contain around 200 base pair of DNA in nucleosome. Okay. H1 protein. 3/4 ton of acidic DNA. Acidic DNA. Three-fourth. Ton. Different nucleosomes are connected with connected. Nucleosomes are connected with each other through linker DNA. Small fragment. Beads on string. B-E-E-D, beads on string. The string. Linker DNA. Different nucleosomes connected with each other. These are nucleosomes. 200 base pair DNA. Wrap up onto this. Okay. Linker DNA. Different nucleosomes are connected with each other through linker DNA. DNA. Small fragment. Fragment. Small fragment. Small fragment converts to solenoid appearance. Solenoid appearance. Solenoid appearance. In higher organisms, arrangement non-histone, role in condensation. Can different functions. Non-histone protein. Histone. In higher organism arrangement, non-histone can have different functions. Role in condensation. Different function. Non-histone protein can have different function. Structural role, regulatory role, RNA polymer, DNA polymer. Also helps in protein. Central dogma. DRP below. Packed to form chromatin fiber that are further coiled and condensed at metaphase stage of cell division to form chromosome. The packaging of chromatin at higher level requires addition set of protein that collectively are referred to as non-histone chromosomal protein. Non-histone. Okay. In higher organism arrangement. The packaging of chromatin at higher level requires additional set of protein. Organism non-histone collectively are referred to as non-histone chromosomal protein. Non-histone chromosomal CHRO chromosomal protein PROTEIN protein. Role in condensation. Okay. The beads string structure in chromatin is packed to form chromatin fiber that are further coiled and condensed at metaphase stage of cell division to form chromosome. The packaging of chromatin at higher level requires addition set of protein called that collectively the packaging of chromatin at higher level requires additional set of protein that collectively are referred to as non-histone protein, chromosomal protein, NHC. In a typical nucleus, some region of chromatin are loosely packed. Rule A. In a typical nucleus, some region of chromatin are loosely packed and stained light are referred to as euchromatin. The chromatin that are more densely packed and stained dark are called heterochromatin. Heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Role in condensation and non-histone chromosomal protein has role in condensation. Okay. Packaging of chromatin at higher level requires additional set of protein. Role in condensation. NHC. Histone chromosomal protein can have different function, structural role, regulatory role, RNA polymerase, DNA polymerase also helps in protein bind. डायरेक्ट सेंट्रल डॉगमा दिया है. भाई साहब, मैं चली बाहर घूमने, मेरा ब्रेन शट डाउन हो गया है, थोड़ी देर में आऊँगी. तुमने विशु को क्यों टाइम आउट दे दिया? वो ना सॉफ्ट टॉपिक की बात कर रहा है। ट्रांसलेशन एक सॉफ्ट टॉपिक है, मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस का ही। उसे पता है कि मैं ये चैप्टर पढ़ रही हूँ ना, इसलिए। चार्जर ही गायब। वो बायो का ही एक, मुझे चार्ज पे लगाना होगा, 5% ही है। जैसे कौन कौन पढ़ रहा है अभी? बी ऑनेस्ट, ओके। अरे हाँ, तू तो पढ़ रहा है, मुझे पता है शुभम। बिसाइड यू। नोनू कहाँ गई? वो भी तो पढ़ रही थी ना हम लोगों के साथ। अब इसमें 25 आ गया भाई, 25 2025 है। मुझे लगा कि वो 24 में। तब तो थी, मुझे भी पता है। अभी है, ये पूछ रही हूँ। सिर्फ एक इंसान पढ़ रहा है, बाकी कोई नहीं पढ़ रहा क्या? या फिर लिख नहीं रहे हो? सो पता नहीं कहाँ से पढ़ रही है। टेलीग्राम पे टेक्स्ट कर सकते हो बुद्धू। छोड़ो, तुम पढ़ो। The Bidzon string structure in chromatin is packaged to form chromatin fiber. Chromatin is packaged to form chromatin fiber that is further coiled and contains chromosome. This is chromosome. CHR chromosome. Okay. This is chromatin fiber. BEADS, beds on string. Beds on string, 200 G speed. Beds on string structure in chromatin. Bidzon string structure in chromatin is packed to form chromatin fiber and further coiled and contains at metaphase stage. The Bidzon string structure in chromatin is packed to form chromatin fiber. Chromatin fiber. Fiber that are further coiled and condensed at metaphase stages of cell division to form chromosome. The packaging of chromatin at higher level requires addition set of protein that collectively referred to as non-histone chromatic chromosomal protein. In a typical nucleus, some region of chromatin are loosely packed and stained lightly and are referred to as euchromatin. Euchromatin loosely packed. PACKED packed. Loosely packed. Lightly stained. Transcriptionally active. Is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Transcriptionally active chromatin. Transcriptionally active chromatin. Transcriptionally active chromatin. Thank you so much, Allah. Chromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Non-histone chromosomal protein is a in a typical nucleus, some region of chromatin are loosely packed. Stained lightly. Heterochromatin. Heterochromatin. Densely packed. Densely packed and stains dark. Transcriptionally inactive chromatin. Transcriptionally TIO inactive. Heterochromatin. Okay. In higher organism arrangement, non-histone protein, chromosomal protein, NHC protein. NHC protein. Non-histone HISTON histone chromosomal CHR. Non-histone chromosomal protein. protein, non-histone chromosomal protein in higher organism. In higher organisms, role in condensation. Of chromatin. Role in condensation of chromatin. And let's see, non-histone chromosomal protein can have different functions: structural role, regulatory role. RNA polymerase, DNA polymerase also helps in protein bind motif structure. पता नहीं क्या लिखना चाह रहे हैं. The beads and string structure in chromatin is packaged to form chromatin fiber that are further coiled and condensed in metaphase stage of cell division to form chromosome. The packaging of chromatin at higher level requires additional set of protein that are packaging of chromatin in higher level requires additional set of protein. Association of H1 protein with a nucleosome indicate the DNA is condensed into a chromatin fiber. H1 with nucleosome. DNA is condensed into a chromatin fiber. This is H1. This is octamer, no? Octamer. Association of H1 and nucleosome. Correspond H1. The DNA is condensed into chromatin fiber. Okay, इसको ना as a whole. pH of histone is basic. The packaging of chromatin at higher level requires additional set of protein. The role in condensation of chromatin. In higher organisms, role in condensation of chromatin. Structural role and regulatory role. Structural and regulatory. Central dogma. It includes the principle of conversion of gene sequence, base pair sequence. Gene expression. Central dogma represents two important steps in our said process. Central dogma. It includes the principle of conversion of gene sequence, base pair sequence into gene expression. Central dogma represents two important steps in above said process. Replication DNA, transcription mRNA, translation protein. Reverse central dogma proposed by Teeman and Baltimore. Decrease proposed by Teeman. Baltimore, also referred to as Teminism, includes the process of reverse transcription. Is restricted in certain virus, retrovirus. restricted method. Okay. Replication of DNA molecule. इतना मुश्किल 15 अभी भी 15 बाकी होगा इतना करने के बाद इतना करने के बाद हमें इतना करने के बाद ये वाला पोर्शन छोड़ेंगे क्योंकि central dogma. It includes the principle of conversion of gene sequence, base pair sequence into gene expression. It includes the principle of conversion of gene sequence, GENE, gene sequence base pair sequence base pair sequence into gene expression. It includes the principle of conversion of gene sequence base pair sequence into gene expression. PRESSION, gene expression. It includes the principle of conversion of gene sequence base pair sequence into gene expression. Central dogma represents two important steps in above said process. It includes principle of conversion of gene sequence base pair sequence into gene expression. Okay, base pair sequence into gene expression. Central dogma represents two important steps in above said process. Transcription. Central dogma represents two important steps, that is transcription and translation. Central dogma, DOGMA dogma, includes two important process. Central dogma includes two important process, है ना बेटा? Two important process. Central dogma represents two important steps. Steps. Transcription and translation. Translation. Reverse central dogma के बारे में डायरेक्टली लिया है, इसमें बताया नहीं है. Okay, the proposition of double helix structure for DNA and its simplicity in explaining the genetic implications became revolutionary. Very soon, Francis Crick proposed the central dogma. Central dogma by Francis Crick. Francis Crick. Okay, by Francis Crick. Proposed the central dogma in molecular biology, which states the genetic information flows from DNA, RNA to protein. DNA, RNA to protein. Okay, central dogma. HIV does not follow central dogma. Reverse transcript is one of me. Replication. होगा DNA होगा ट्रांसक्रिप्शन के थ्रू. TRANS ट्रांसक्रिप्शन. CRIPTION ट्रांसक्रिप्शन. RNA, mRNA, protein, protein, translation, transcription, translation, mRNA देखिए protein, okay. HIV do not follow central dogma. HIV, retrovirus, this is central dogma, CENTRAL, central, DOGMA, dogma. Retrovirus does not, Hastak feral law includes the process of reverse transcription, is restricted in central virus. Retrovirus includes the process of reverse transcription It is restricted in certain virus. Retrovirus, okay. Retrovirus does not follow central dogma. CENTRAL, central dogma. Reverse central dogma proposed by Temin and Baltimore. ये सब तो नहीं दिया है ना एनसीईआरटी में. In some viruses, the flow of information is in reverse direction, that is RNA to DNA. Can you name this? Reverse transcription, okay. Reverse transcription, Teminism. Teminism, TEMI, Teminism. Temin and Baltimore मत पढ़ो ये सब, एक्स्ट्रा हो गया ना एनसीईआरटी से. Includes the process reverse transcription, okay. Certain virus, restricted to certain virus, 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 like retrovirus, retrovirus, okay. Retrovirus. Okay, that is RNA to DNA. RNA to DNA bananes, okay. mRNA. Reverse central dogma. Reverse central dogma. The reversal central dogma refers to the process where genetic information flows from RNA to DNA. Okay, other RNAs. Transcription, DNA replication. क्या दिखा रहा है ये सब? Central dogma of molecular biology, DNA, replication, transcription. Okay, replication. Replication को भूलना नहीं है भाई. DNA replication, transcription, mRNA translation, protein. This is central dogma. पता नहीं. RNA DNA बनेगा. अच्छा, RNA पहले DNA में बनेगा, DNA. फिर यहाँ पर रेप्लिकेशन, REPLI, रेप्लिकेशन होगा. उसके बाद यहाँ पर होगा ट्रांसक्रिप्शन. ट्रांसक्रिप्शन होके ना यहाँ पर mRNA बनेगा. उसके बाद जाके यहाँ पर ट्रांसलेशन होगा. प्रोटीन PROTEIN यही होगा ना। रिवर्स ट्रांसक्रिप्शन किनेज़। छोड़ो, इससे ज़्यादा नहीं पढ़ना है बस। Replication, duplication of DNA molecule. Replication. Duplication of DNA molecule. It includes mainly DNA-dependent DNA polymerase. It includes mainly DNA-dependent DNA polymerase. Important है भाई, रेड से लिख लेती हूँ। Replication, DNA-dependent DNA polymerase. Polymerase included in replication replication DNA dependent DNA polymerase, very important. Replication, okay. DNA dependent DNA polymerase. It includes mainly DNA dependent DNA polymerase. Need a template. DNA dependent DNA polymerase needed a DNA template based on the principle of complementarity. Polymerization of new strand of DNA is observed. Okay. DNA dependent DNA polymerase DNA dependent DNA polymerase need a DNA template. Needs a DNA template. DNA template based on principle of complementarity. Based on the principle of complementarity. Based on the principle of complementarity. Needs a DNA template. DNA dependent DNA polymerase needs a DNA template. Polymerization of new strands of DNA is observed. DNA template needs a DNA template. DNA dependent DNA polymerase needs a template. Polymerization on the basis of complementarity. Based on the principle of complementarity. Polymerization of new strand is observed. Polymerization of new strand. Polymerization of new strand is observed. Polymerization of new strand is observed. बाहर में जाए क्या? Polymerization of new strand DNA is observed. First one universal DNA dependent DNA polymerase. Then the template strand signs. Template strand. Third point Q. On the basis of the principle complementarity. कुछ भी. शायद. दिखता तो है। बायोटेक आज तो गया था भाई साहब, 7 घंटे में भी कुछ नहीं हुआ। DNA dependent DNA polymerase need a DNA template based on the principle of complementarity. Polymerization of new strand, DNA strand, यहाँ पर DNA लिखो बहन चुपचाप, DNA strand is absorbed. Polymerization of new strand of DNA is absorbed. DNA strand is absorbed. Okay. In prokaryote, replication is observed at once. That means it starts at one end of DNA segment and continuously fulfill the duplication. Continuously fulfill the duplication until the end of DNA is observed. Single start point origin of replication. Single start point origin of replication. Only one ORI is found in prokaryotes. Okay, because DNA is single and circular. In eukaryotes, origin of replication is observed at many sites. Bhai sahab. Process of replication. Based on the DNA polymerase, many other enzymes are involved. Based on DNA polymerase, many other enzymes are involved. Enzyme involved DNA polymerase. In prokaryotes, mainly 3 types of DNA polymerase. DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Function. Highest rate of polymerization is observed in DNA polymerase at 2000 base pair per second. All the DNA polymerase do polymerization in 5' to 3' direction. 5' to 3' direction. ये भी important है PYQ के point of view से. DNA polymerase also helps in proofreading for correction and accuracy. DNA polymerase can't do de novo synthesis or new synthesis. De novo, a new synthesis. It always requires short fragment of RNA primer to polymerize the new DNA. DNA polymerase also helps in proofreading for correction and accuracy. Exonuclease activity is observed generally in 3' to 5' direction. But 5' to 3' exonuclease is observed by DNA polymerase 1 is done to exercise exist. Disconnect the primer. क्या लिखा है मैंने? $28. Replication is observed in prokaryotes. Observed at one. Means replication is observed at once. Single start point. Single start point or origin of replication. Hi. Only one ORI is found in prokaryotes. Replication is observed at one single starting point or ORI of origin of replication. Only one ORI is found in prokaryotes. It starts at one end of DNA segment and continuously fulfill the replication until the end of DNA is observed. Continuously fulfill the replication until the end of the DNA is observed. DNA is single and circular. DNA is single and circular. Okay. It starts at one end of DNA segment. Continuously fulfill the replication until the end of DNA is observed. Starts replicating. Starts replicating. Starts replicating from one end of DNA and continuously continuously fulfill the replication. Continuously fulfill the replication until the end of DNA is observed. It starts replication at one end of DNA segment and continuously fulfill the replication until the end of the DNA is observed. Single starting point, origin of replication, ORI. Only one ORI is found in prokaryotes because DNA is single and circular. DNA is single and circular. Single and circular. DNA is single and circular. In eukaryote, origin of replication is observed at many sites. In eukaryotes, EUKARYOTS. In eukaryotes, origin of replication is observed at many sites. Origin of replication. Origin of replication. Origin of replication, ORI. Origin of replication is observed at many sites. Many sites. Observed at many sites. Process of replication. Beside DNA polymerase, many other enzymes involved. DNA polymerase. Okay, process of replication. Beside DNA polymerase. Beside the DNA polymerase, many other enzymes are involved. Enzymes are involved. Involved. Beside DNA polymerase, many other enzymes are involved. Enzyme involved. DNA polymerase. Beside DNA polymerase, many other enzymes are involved. Enzyme involved. DNA polymerase. Enzyme involved. DNA. What are the enzymes that are involved in the process of replication other than DNA polymerase?
Besides DNA polymerase, the key enzymes involved in DNA replication include helicase, unzips DNA, primase, synthesizes RNA primers, ligase, joins DNA fragments, and topoisomerase, relieves DNA supercoiling. Here's a more detailed explanation of each enzyme's role. 1. Helicase. This enzyme
What are the enzymes that are involved in the process of replication? Beside DNA polymerase, many other enzymes are involved. Helicase, primase. Helicase unzips DNA. Primase synthesizes RNA primer. Ligase joins DNA. Ligase joins DNA fragments. Synthesize RNA primer. Primase synthesize RNA primer. Helicase and topoisomerase. Topoisomerase. MERASE receives DNA supercooling. Topoisomerase. Khan Academy. Process of replication. Beside DNA polymerase. Process of replication. Beside DNA polymerase, many other enzymes are involved. Enzymes involved. Okay. function. Highest rate of polymerization is observed in DNA polymerase. In prokaryotes. In prokaryotes, mainly 3 types of DNA polymerase. Found DNA polymerase in prokaryotes, mainly 3 types of DNA polymerase. DNA polymerase 3. Highest rate of polymerization is observed in DNA polymerase 2000 base pair per second. Polymerase 2 polymerization. Okay. Polymerize. Highest rate of polymerization. Highest rate of polymerization is observed in DNA polymerase 2000. All the DNA polymerase do polymerization in 5' to 3' direction. Highest rate of polymerization is observed in DNA polymerase 2', 2000 base pair per second. Highest rate of polymerization is observed in DNA polymerase. DNA polymerase also have been proofreading. Okay. Highest rate of polymerization is observed in DNA polymerase 2000 base pair per second. All the DNA polymerase do polymerization in 5' to 3' direction. Bacterium DNA polymerase 1, DNA polymerase 2 र DNA polymerase 3 हुन्छ. DNA polymerase also helps in reading for correction and accuracy. Proofreading नै त भन्छ यसलाई नै. Exonuclease activity is observed generally in exonuclease activity चाहिँ 3' to 5' direction मा हुन्छ, but 5' to 3' exonuclease is exonuclease is absorbed by DNA polymerase 1. क्या लिखा है? By DNA polymerase 1 is done to exercise exist disconnect the primer. DNA polymerase can't do a new synthesis, de novo synthesis. Okay, सारे 4 पॉइंट से लिख देते हैं. ये रहा 1 point, ये रहा 2 point. This is 3rd point. Highest rate of polymerization is observed in DNA polymerase 2000 base pair per second. All the DNA polymerase do polymerization from 5' to 3' direction. ये तक किया है मैंने. Polymerase, many other enzymes are involved. Okay, DNA polymerase also helps in proofreading for correction and accuracy. Also helps in proofreading. Proofreading, REA. DNA polymerase also helps in proofreading for correction and accuracy. DNA polymerase also helps in proofreading for correction accuracy. For correction, CORREC. Correction and accuracy. Accuracy. DNA polymerase also helps in proofreading for correction and accuracy. DNA polymerase also helps in proofreading, correction and accuracy. DNA polymerase also helps in proofreading for correction and accuracy. DNA polymerase can't do de novo synthesis. DNA polymerase can't do de novo synthesis. A new synthesis. De novo synthesis. DNA polymerase can't do de novo synthesis. A new synthesis. It always requires a short fragment of RNA. This requires a short fragment of RNA primer to polymerase. RNA primer. DNA polymerase can't do de novo synthesis. It always requires a short fragment of RNA polymerase. RNA polymerase to polymerize the new DNA. RNA primer. A short fragment of RNA polymerase. Okay, it always requires a short fragment of RNA. RNA primer. It always requires a short fragment of RNA. RNA primer to polymerase the new DNA. RNA primer to polymerase. To polymerase the new DNA. To polymerase the new DNA. DNA polymerase can't do de novo synthesis. New synthesis, okay. It also requires a short fragment. It also requires a short fragment of RNA primer to polymerize. To polymerize, लिखा है polymerize, RISE. To polymerize the new DNA. It also requires a short fragment of RNA primer. RNA primer to polymerize the new DNA. To polymerize the new DNA. Okay, DNA polymerase also helps in proofreading. Proofreading के लिए. Proofreading. DNA polymerase also helps in proofreading. DNA polymerase proofreading. प्रूफ रीडिंग। भाई साहब भाड में जाए सब कुछ। रिडिङ प्रुफ रिडिङमा के हुन्छ कुनी त। Proofreading. Proofreading. Helps in proofreading. Exonuclease activity. Exonuclease activity. Exonuclease activity is observed. DNA polymerase also helps in proofreading. Proofreading. Exonuclease activity is observed generally. In 3' to 5' direction. 3' to 5' direction. In 3' to 5' direction. Exonuclease activity is observed generally in 3' to 5' direction. But 5' to 3' exonuclease is also observed. But 5' to 3' exonuclease is observed by DNA polymerase 1. Exonuclease activity is observed generally in 3' to 5' direction. But 5' to 3' exonuclease is observed by DNA polymerase. Exonuclease, nuclease, 5' to 3', exonuclease, CLEASE. Generally 3' to 5' exonuclease activity. 5' to 3' exonuclease is observed by OBSER, observed by, I feel so bad for them, is observed by DNA polymerase 1. DNA polymerase 1. Astaghfirullah. DNA polymerase polymerase is absorbed by DNA polymerase 1. Done to exercise, disconnect the primer. DNA polymerase 1. DNA polymerase 1 is done to exercise. Okay, DNA polymerase 1. DNA polymerase 1 exercise, disconnect. Done to disconnect the primer. Disconnect, CONNECT, disconnect the primer. Slot 7, slot 7. DNA polymerase. Helix. Exonuclease activity is observed generally in 3' to 5' direction, but 5' to 3' exonuclease is observed by DNA polymerase 1, and it is also used for disconnecting the primer. Okay. All the DNA polymerase do polymerization from 5' to 3' direction. All the DNA polymerase do polymerization in 5' to 3' direction. Highest rate of polymerization is observed in DNA polymerase 2000 base pair per second. 2000 base pair per second. Highest rate of polymerization is observed in DNA polymerase 2000 base pair per second. DNA polymerase also help in proofreading. Helps in proofreading for correcting and accuracy. DNA polymerase also helps in proofreading for correction and accuracy. DNA polymerase can't do de novo synthesis, new synthesis. It always requires a RNA primer, short fragment of RNA primer to polymerize the new DNA. It always requires a short fragment of RNA primer, RNA, to polymerize the new DNA. DNA polymerase can't do de novo synthesis, new synthesis. It always requires a short fragment of RNA primer, RNA, to polymerize the new. यार अपना रोज ऐसे ही लॉन्ग सेशन लगाया करो, आपको पढ़ता हूँ मैं. ओके साइद, अब पढ़ाई करो, कुछ भी मत लिखो वहाँ पर. DNA polymerase can't do de novo synthesis. It always requires a short fragment of RNA primer, RNA, to polymerize the new DNA. Proofreading. Exonuclease activity is observed generally in 3' to 5' direction, but 5' to 3' exonuclease is observed by DNA polymerase 1. Proofreading. Exonuclease activity is generally in 3' to 5' direction, but 5' to 3' direction is also done by polymerase 1, DNA polymerase 1. Exonuclease, okay, 5' to 3', exonuclease is observed by DNA polymerase 1. DNA polymerase 1 is used to disconnect the primer. Helicase, कहाँ पर है 1? Helicase. DNA polymerase, ये सब है DNA polymerase. हेलिकेस, उसके बाद प्राइमेस की तो बात ही नहीं है तो वहाँ पर DNA लाइगेस की भी बात की है. It helps in unwinding the DNA that exists. Now, it helps in unwinding the DNA that results into replication fork. Helps in unwinding. HELPs helps in unwinding. Unwinding. WINDING. Unwinding. It helps in unwinding of DNA. Astaghfirullah. It helps in unwinding of DNA. Results into unwinding of DNA. अस्तगफिरुल्लाह, मुझे बहुत ही खराब लग रहा है. Helps in unwinding. Helps in unwinding of DNA that results into replication fork. Replication that results into replication fork. FORK fork. Unwinding. Helix helps in unwinding of DNA that results into replication fork. Replication fork. The single strand binding protein SSBS. Single strand binding proteins helps in maintenance of single strand of replication. Okay, SSBS अंदर से ले. क्या है? It prevents complementarity. It prevents complementarity after unwinding. अच्छा. Topoisomerase. It relieves the strain in DNA. It helps in preventing positive supercoiling. In prokaryote known as DNA virase. Primase helps in formation of RNA primer. DNA ligase helps in binding or attaching short fragment of DNA. पता नहीं क्या क्या आया. Helicase. It helps in unwinding the DNA that results into replication fork. It helps in unwinding of DNA. Helicase. Unwinding of DNA. Replication fork. Single strand binding protein SSBS. SSBPS. Single strand binding protein. Proteins. 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 Single strand binding proteins. DNA ligase तक करो ना, फिर उसके बाद देखा जाएगा. Single strand binding protein SSBPS helps in maintenance of single strand. Maintenance. MAI. Maintenance of single strand for replication. Helps in maintenance of single strand for replication. Helps in maintenance of single strand for replication. Helps in maintenance of single strand. Helps in maintenance of single strand for replication. It prevents complementarity after unwinding. It prevents complementarity after unwinding. It prevents complementarity after unwinding. Isomerase. Relieves the strain in DNA. It relieves the DNA strain in DNA. It helps in preventing positive supercoiling. Strain. It relieves the strain in DNA. It relieves the strain in DNA. It relieves the strain in DNA. Helps in preventing positive supercoiling. Helps in preventing PREVENT. Helps in preventing positive supercoiling. Supercoiling. COILING. Supercoiling. It helps in preventing positive supercoiling, known as DNA. Okay. So oops. क्या बोल सकती थी मैं? कुछ नहीं बोल सकती थी, इसलिए मैं चुप ही रही था. Also known as DNA gyrase in prokaryotes. It is a strain in DNA. It relieves the strain in DNA. Helps in preventing the positive supercoiling. Helicase single strand. SSBS बाइंडिंग प्रोटीन 1 2 3 4 2 पॉइसमरेज 4 प्राइमेज कौन लिखेगा बहन? PRIMAS आलू के चश्मा लगा के भी नहीं दिखता. प्राइमेज हेल्प्स इन फॉर्मेशन ऑफ RNA प्राइमर. प्राइमेज हेल्प्स इन फॉर्मेशन ऑफ DNA ligase. हेल्प्स इन बाइंडिंग और अटैकिंग. कितना ज़्यादा प्यास लग रहा है ना मुझे. हेल्प्स इन बाइंडिंग BIN. हेल्प्स इन बाइंडिंग और अटैचिंग शॉर्ट फ्रेगमेंट ऑफ DNA. हेल्प्स इन बाइंडिंग और अटैचिंग शॉर्ट फ्रेगमेंट ऑफ DNA. SHORT शॉर्ट छोर बहन छोर FRAG फ्रेगमेंट शॉर्ट फ्रेगमेंट ऑफ DNA. Helps in binding or attaching short fragment of DNA. Helps in binding or attaching short fragment of DNA. DNA polymerase को different functions छ. Okay, proofreading छ. 5' to 3' direction मा polymerization गराउँछ. त्यहाँदेखि एक्सोन्युक्लियस को युजुअली 3' to 5' direction हुन्छ. तर यसले चाहिँ polymerase 1 ले के गर्छ? 5' to 3' direction मा polymer रिमुभ गर्छ base pair लाई nitrogenous base level and DNA polymerase 1 is used to disconnect the primer. Okay. नयाँ प्रोटिन नयाँ स्ट्रान्ड सिन्थेसाइज गर्न सक्दैन DNA ले. RNA प्राइमर चाहिन्छ. RNA को सानो फ्रेगमेन्ट चाहिन्छ. 2000 बेस पेयर पर सेकेन्ड गर्छ हाइएस्ट पोलिमराइजेसन चाहिँ. भयो. हेलिकेसले के गर्छ भन्दा त अनबाइन्डिङ हेलिकेस अनबाइन्डिङ अफ द DNA स्ट्रान्ड. The result in replication for SBS. Single stranded binding protein ले चाहिँ further complementary base pair लाई जोडिनु दिँदैन. Helps in maintaining a single strand for replication. It prevents complementarity after unwinding. Topoisomerase positive supercoiling हुनुपर्दा बचाउँछ. DNA gyrase पनि भन्छ प्रोकार्योट्समा टोपोआइसोमरेजलाई. DNA gyrase helps in preventing positive supercoiling. Primase RNA प्राइमर बनाउनमा हेल्प गर्छ. DNA ligase जोडाउँछ दुईटा DNA फ्रेगमेन्टलाई. हेल्प्स इन बाइन्डिङ अर अट्याचिङ सर्ट फ्रेगमेन्ट अफ DNA. मैं पानी लेके आई, ओके? I'm thirsty. मग भी चाहिए क्या? लगता तो है। है टोपोआइसोमरेज पोजिटिभ सुपर कोइलिङ हुनुबाट बचाउँछ डिएनए गाइरेज भन्छ प्रोकार्योटमा replication both strands are involved in replication. Okay, polymerase 5' to 3' replication needs the primer. The DNA polymerase 1 RNA primer is removed with the help of DNA polymerase 1. Topoisomerase positive supercoiling हुनुबाट बचाउँछ DNA ligaseले जोडिन्छ सँगसँगै DNA fragmentलाई. Many short fragments of RNA primer observed by Okazaki, so referred as many short fragment of RNA primer observed by Okazaki, so referred as Okazaki fragment. This continuous strand, एउटा continuous strand भन्छ. Okay, continuous strand लाई नै leading strand पनि भन्छ. Discontinuous strand लाई lagging strand. Polymerase 5' to 3' direction of polymerization. Polymerase it needs the primer. Okay, 5' poly free phosphate PO4 लेखेको होला यो semi-conservative DNA replication. नीड्स प्राइमर 5 प्राइम टू 3 प्राइम पॉलीमराइजेशन और रेप्लिकेशन। भाई साहब नहीं होगा मुझसे ये सब, दुख दर्द। Five prime to 3 prime. 5 prime to 3 prime. DNA polymerase requires 3 prime OH provided by RNA primase. समझ में तो नहीं आ रहा है भाई साहब मुझे. 5' to 3', again 5' to 3'. Okazaki fragments are absorbed. Okazaki fragment. Fragments observed by Okazaki, so referred as Okazaki fragment. Short fragment of RNA primer. Fragment are present. DNA primer. Many short RNA primer fragment. RNA primer fragments are absorbed. Many short Okazaki fragments are present. Many short RNA primer fragments are absorbed. Many short RNA primer fragments are absorbed. Ligase, DNA, ligase, it's the small fragment. Fragments. DNA ligase. Polymerase 3' OH provided by RNA primase. End for polymerization. Three prime to 5'. 5'. 5' to 3'. 5' to 3' again. DNA polymerase requires 3' OH. 3' OH. OH provided by RNA primase. Primase or primer, free end for polymerization. Polymerase requires 3' OH provided by RNA polymerase. Free end for polymerization. Free for polymerases. Polymerization. HP phosphate 3 prime OH. अभी किस चीज़ के लिए अलार्म बज रहा है? Semiconservative observed contain one strand of parent DNA and the other strand of newly polymerized synthesized. Okay. Contain one strand of parent DNA. Contain contain one strand of parent DNA and the other strand of newly polymerized daughter DNA. Keep a daughter DNA or I need eraser. DNA. This mode of replication is called semi-conservative mode of replication, was observed by Michelson and Stoll. Observed by semi-conservative DNA. Okay, observed by observed by Michelson, and Stoll. STAHL Stoll. Michelson and Stoll in E. coli. Later, Taylor Visiafava. Taylor and colleague. Fava beans. 1958. Visiafava. Fava beans. Okay. Michelson Stoll used carbon and chloride gradient technique. Carbon and chlorine gradient. नहीं पता. शेरी के लिए लोग उठ गए शायद. Calcium chloride gradient. Calcium chloride नहीं, cesium and chlorine gradient. Cesium chloride ये कि and लिखा है CSCl लिखना चाहिए था ना मुझे. CSCl. Cesium chloride gradient to measure. Cesium chloride gradient to measure. Cesium chloride gradient technique. Okay. Cesium chloride gradient technique. Michelson and Stoll. Michelson Stoll used cesium chloride gradient technique. Taylor used radioisotope thymidine technique. Taylor used radioisotope thymidine technique. Thymidine technique. TECHNIQUE technique. Good luck शकुंतला ना संजुक्तला. संजुक. मेरे आंखों के चश्मे का पावर और बढ़ाना पड़ेगा. संजुक्ता. RNA world. You mean right? Transcription. Taylor used radioisotope thymidine technique. Cesium chloride. अरे ये Cl है. density gradient, cesium chloride density gradient, right? Cesium chloride density gradient, cesium chloride density gradient, thymidine, radioactive thymidine. H3. Cesium chloride gradient in missiles and stuff. स्टॉप प्लेइंग डम्ब कन्हैया, यहाँ पर लिखा तो है साफ साफ. आई थिंक यू कैन रीड है ना. N15 isotope medium. Isotope. E. coli is initially cultured grown in N15 isotope medium. Result into the corporation of N15 in DNA. Result into the corporation of N15 in DNA. Contains NH4Cl. Contains N15 H4Cl. NH4Cl N15. At nitrogenous base. As nitrogenous base is pair of DNA. Nitrogenous base is pair of DNA. E. coli subsequently cultured in N14. N15 होना चाहिए ना. N15 isotope medium. The E. coli frozen reproduction had been used at specific time interval 20 minutes, 40 minutes, 60 minutes, 20, 40 and 60. बहुत मटकी कर रही हूँ ये सब. क्या दिखा रहा है? इतना तो करना ही होगा, उसके बाद ही करना पड़ेगा. E. coli is initially cultured grown in N15 isotope medium. It is PYQ stability H bond. It is now proven that DNA replication semi-conservatively. It was shown first in E. coli and subsequently in higher organisms such as plants and human cells. Matthew Michelson and Franklin Stahl performed the following experiment in 1958. They grew E. coli in a medium containing N15, NH4Cl. N15 is the heavy isotope of nitrogen, as the only nitrogen source for many generations. The result was that N15 was incorporated into new synthesized DNA as well as other nitrogen containing compounds. The heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride density gradient. Please note that N15 is not a radioactive isotope and it can be separated from N14 only based on densities. Then they transferred the cell into a medium with normal. Into a medium with normal NH4Cl and took sample at various definite time interval. Medium with normal NH4Cl. Then they transferred the cell into a medium with normal NH4Cl and 14 and took sample at various definite time interval as the cell multiplied and extracted the DNA that remained as double-stranded helices. The various samples were separated independently on cesium chloride gradient to measure the density. Coli is initially cultured. E. coli is initially cultured, grown in N15 isotopes medium. Into incorporation. NH4Cl contain NH4Cl as nitrogenous base in इतने मैनरली स्लो कहते हैं ना, क्या ही बोलूं. कुछ लोग सेरी के लिए उठ गए हैं ना, लेकिन इतना ज़्यादा थूक रहे हैं। बेसिक मैनर सब में होना चाहिए यार। वो भी इतने जोर जोर से थूक रहे हैं, चॉकलेट जैसे चाटा ही लगा दो। Cultural chromin N15 isotopes may give resulting into incorporation of N15 in DNA. NH4Cl as nitrogen base as nitrogenous base is pair of DNA. क्या लिखना चाह रही हूँ मैं, मुझे नहीं पता। NH4Cl N15 equal is subsequently cultured in N14 isotope medium. N14 N14 medium NH4Cl. Astaghfirullah. The E. coli progeny reproduction had been observed at a specific time interval 20 minutes, 40 minutes, and 60 minutes. As subsequently the E. coli progeny okay, had been observed. I'll go up, okay, I'll sleep. Had been observed at a specific time interval. Specific. या फिर मुझे नींद नहीं आया तो जाने के बाद, अभी तो आ रहा है इंटरवल। E. coli progeny reproduction had been observed at a specific time interval 20 minutes, 40 minutes, and 60 minutes. Okay. What could be the percentage possibility of lighter pentagon 15 DNA after 60 minutes? 15 minutes. Good night, fish. Bro, but I'm not sure कि मुझे नींद आएगी कि नहीं। अभी आ रहा है, ऐसा लग रहा है कि मेरा दिमाग बंद हो गया है, लेकिन मैं श्योर नहीं हूँ। उधर जाने के बाद मुझे ना भी आ सकते हैं। अच्छी तरीके से ख़त्म किया ना, थोड़ा थोड़ा बचा है बीच में। मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस कल ख़त्म होगी यार। सिर्फ 7 स्लॉट ही कंप्लीट हुआ, 1 तो बच गया। वैसे ये ना स्लॉट ना कंप्लीट हो गया होगा। मैंने बीच बीच में टाइमर ऑन करना भूल गई थी, पॉज करके। तो श्योर नहीं हूँ। गुड नाइट ऑल। वैसे मैं कोशिश करूँगी सोने की, नींद नहीं आई तो वापस आ जाऊँगी। गुड नाइट संजुक्ता। कोशिश करेंगे, बहुत अच्छी नींद आए तो अच्छी तरीके से सो जाऊंगी। या अल्लाह अच्छा नींद आए। हाँ मस्त ही सोएंगे, सोने में कोई मुझे टक्कर दे सकता है क्या? चलो ट्राई करती हूँ। गुड नाइट एवरीवन, बाय बाय। नींद अच्छे से आया ना अभी, तो मैं वो लाइव भी ऑफ कर दूंगी। नहीं पहले ट्राई ही करती हूँ ना लाइव ऑफ करके। लाइव ऑफ कर रही हो ओके। बाय बाय। बाय शुभम, बाय नोनू, बाय एलेक्स, बाय श्री, बाय संजुक्ता, और कौन रह गया? बाय आदर्श।